There we go. So the content on the exam should get easier as we continue to move forward, but also your familiarity with how to take an exam should improve as well. The exams will be released today. You'll have about 24 hours to look at them and see where you got your points and where you didn't get your points, and what kind of things, and try to figure out what kind of things you could do to improve that. And if you're not sure, then by all means, come to my office hours, email me so we can discuss ways that you can improve. So that's a good thing, that the test should, you should, if not easier, you should get more comfortable with them, getting better at anticipating what kind of questions are going to be on there, and getting better at how to write your essay questions. You have to remember, there are two things you need to have to be successful in a class like this. Yes, you have to cram a large amount of information into your brain, but you also have to be able to put that information on the page. And writing an essay question is just like shooting a basketball. It is a skill, and not everybody is equally competent at it. However, everyone with practice can get better. And so if you're struggling with that part of this, then that is something you can continue to work on, and I would be happy to help you with as well. The good news is, like I said, the lab and lecture exams are not the only things that determine your grades. All of your points count in, and you guys did excellent on your group presentation, so that was excellent to see. And so the good news is, overall, we've got a 76% in the class right now. But again, I would like to continue to see that go up. There's no reason that our average overall in the class shouldn't be above 80. So easily half of the students should be getting A's and B's on this. So again, we're only about halfway through the points, a little less than halfway through the points. Like I said, we'll probably finish somewhere around 12 to 1,300 points in this class. So there is some wiggle room still. Remember, if you struggled on one of the first two lecture exams, remember your final exam can replace your lowest lecture score. So if you totally bombed a lecture score, if you got a 14 on a lecture exam, then you can replace that with an 84 if that's what you get on your finals. So there is still a lot of wiggle room on this to help you to be successful. Now, let's talk about today's material. The good news is we're just doing one organ system. It's very simple. It's very straightforward. However, there are a couple challenges to this section. The first is that this is typically 3.5 days worth of lecture. And depending on the semester, some semesters, because the way the holidays fall, we have 4 days for this section. And sometimes, like we do this semester, we only have 3. So the nice thing is that if we were in the classroom, we'd be doing more hands-on activities. We're not going to be able to have that opportunity, but that does mean a little bit more talking during lecture. So we have a lot of material to cover on each of the 4 days. Sorry, the 3 days, to cram 3.5 days worth of information into it. The other thing I want to point out to you is that it's a quick turnaround with this one section. It's 2 weeks from the last exam. You only have 2 weekends to study for this material, and one of them is already passed. So you only have 1 more weekend to study for this material, and we have a Thursday exam again. It's not ideal, but again, it's the way that these classes have fallen out. So we have that Thursday exam, which means 2 things. It means that this coming weekend is your big weekend to study. You are getting presented with new material on Tuesday, and it'll be a lot of new material, but you've got the outlines, you've got your textbook and your other resources. You've got a couple lab simulators that should help you to understand this material, to help you to be successful on that. So you'll have a busy weekend that way. And then also, just like we did this last time with the Thursday exam, to encourage you to start looking at the new material ahead of time, I will be assigning a couple pre-labs. I'm not going to worry about the page numbers or any of that stuff yet. I just want that out there in the back of your consciousness, so that as we get closer to next week's exam, you remember that after Thursday's exam, you don't get to just party for 5 straight days. You'll have to at least give me 2 hours of work to do the 2 pre-labs that you're going to be responsible for. And hopefully you'll do more than that as well. All right, that is the game plan. Any questions on any of that? Excellent. You know how much I love my stunned silences first thing in the morning. Spectacular. All righty. Then with that, let's dive into the digestive system. Digestive system again, like all organ systems, is comprised of two main groups of organs. We have the primary organs and the accessory organs. And of course, the primary organs in the digestive system are what are known as the gastrointestinal tract, or also known as the alimentary canal. This is basically a hollow tube that runs all the way from your mouth to your anus. And there are two important things we know about this. Let's start about one that we have probably talked about already, and that is, what do we know about the lumen of the digestive system? My microphone is on, right? Words are coming out of my mouth.
Yeah.
Okay.
It's lined with villi.
Okay, is the entire lumen of the digestive system lined with villi or microvilli?
No.
No, there are portions of it that are, absolutely, but not all of it. What is something that that we know about the entire lumen of the digestive system?
It has a mucosa.
True, absolutely. It is a mucosa because why? Why would it be lined with a mucosa?
Because it's open to the outside.
It is indeed lined with a mucosa because it is open to the outside world. And in fact, not only is it open to the outside world, but the lumen of the digestive system is the outside world. Did we not talk about this already? I thought we had talked about this before. Where's my penny? Oh, here, I'll use my fidget. Excellent. Here's my little fidget thing. If I hold this in my hand, is it inside my body?
No.
If I wrap my fingers around it, is it inside my body?
No.
No, it may be surrounded by my body, but it is not inside my body. To get inside of a body, you have to pass through a membrane. So instead, if I took this and held it inside my mouth, is it inside my body?
No.
But what if I closed my mouth and swallowed it? Would it be inside my body then?
Yeah.
No,
No.
once
No.
again, it would still just be surrounded by my body. Hopefully nobody's swallowing fidget toys, but there are plenty of little kids that swallow pennies. And when that kid swallows a penny, what happens to that penny? Is it gone forever?
They'll eventually pass it.
Yeah, in most cases, that penny is going to come out the other side. A little cleaner or a little dirtier, depending on how you think of it. Right? And at no point in that process did that penny enter that kid's body. Right? However, that kid may have had a cheeseburger for breakfast, like all good kids should. Right? And when that cheeseburger is swallowed into our alimentary canal, it's broken down into bits and pieces. And then those bits and pieces cross the membrane of our alimentary canal and enter inside of our body. We absorb it into our body, right? Our digestive system, our body has no needs internally of currency. There are no currency receptors, so that penny passes right through, but the pieces of the cheeseburger are absorbed. They cross the membrane. As you may or may not recall, we have a fancy name for this. Because the lumen of the digestive system is considered to be outside of the body, we call it the topological exterior. I thought we had talked about this because remember when we were talking about the difference between exocrine and endocrine glands, we talked about how things like your salivary gland or how things like the glands that produce the hydrochloric acid in our stomach are examples of exocrine glands. Exocrine glands, of course, use ducts to release stuff outside the body. So releasing those secretions into the lumen of our digestive system is considered the outside of the body. All right, so we have this hollow tube. In the cadaver we have in the classroom, which if we were in the classroom, you would have had the opportunity to see that alimentary canal, that hollow tube from mouth to anus, is about 30 feet long. Now, why do I have to qualify that in the cadaver? Don't you all have alimentary canals? Not a trick question. These are the
Yeah.
easy questions.
Because it's
Yes.
relaxed in
I'm
the
sorry?
Is it because it's relaxed in the cadaver?
Exactly. Death is many things, and one of them is indeed relaxing. So while all of you have an alimentary canal, you have a fair amount of muscle tone in your alimentary canal. So your alimentary canal is much, much shorter than 30 feet in length. However, when the body dies and those muscles relax, it extends out to its full 30-foot length. All right. So all of the main organs, or all of the organs that food passes through, right, makes up the alimentary canal. So again, that would be like your mouth, like your esophagus, like your stomach, your small intestine, large intestine, things like that. However, there are organs that food doesn't pass through, and those would be the accessory organs. Give me some examples of some of the accessory organs of our digestive system.
Liver,
Liver, excellent. Food doesn't pass through
Stomach.
that. I'm sorry?
stomach,
Gallbladder. Well, remember, stomach food passes through the stomach. So the stomach is part of that hollow tube, but the gallbladder, the liver,
The tongue?
teeth, absolutely. It goes over, or if you have a big gap between the teeth, but it doesn't go through the tooth, right?
What
It
about
goes-
salivary glands?
Salivary glands, pancreas, absolutely. Tongue, right? Again, food may go over the tongue. The tongue may move food around, but I guess unless you have a really big stud and you take out that piercing, then maybe food can pass through your tongue, but otherwise it's part of the accessory organ. So yeah, basically the easiest way to tell whether something is an accessory organ or part of the alimentary canal is whether or not food passes through it. All right, questions on that? All righty. Let's talk about the functions of the digestive system. What are the big functions of the digestive system? Give me one of the functions.
A chemical breakdown of food?
Excellent. One of the functions, not surprisingly, is digestion. However, as was mentioned, we can break digestion down into a couple ways. Digestion just means the breakdown of food. But as you guys have pointed out, we can do that one of two ways. Chemical digestion, that's not how you spell chemical. And as Amy pointed out, also mechanical digestion. What's the difference between the two?
Force versus reaction?
Oh, I like that. Absolutely. Mechanical digestion definitely involves a force. Basically, this is just a breakdown of big things into little things. If I have a boulder and I have a hammer and a chisel, I can break that one rock down into many little rocks. But I haven't changed the composition of the rock when I've done that. I may have changed its shape, I may have changed its surface area, but I haven't changed the composition of it. To change the composition of it, as you pointed out, we need to break bonds. And when we break bonds, we change the chemical composition of the components. So we take that glycogen and we break it down, or that starch, and we break it down into individual glucoses. We break proteins down into amino acids, right? We break bonds to take macromolecules into their basic building blocks. Excellent. Perfect. I like that. All right. What else? What's another function of the digestive system? Absorption. Excellent. Absolutely, absorption. Again, the absorption is the process of bringing the resources that we want or need or that can get in past that mucous membrane and into the body. So passing that mucous membrane, getting into the body, that is absorption. Excellent. What else?
Well, we got to get it out.
Absolutely. The things that we don't absorb leave via what process? There we go. Defecation. Absolutely. This is anatomy and physiology class. You are allowed to say defecation, right? If you can't handle the digestive system, just think how awkward the reproductive system is when you're gonna have to say penis and vagina, right? So yeah, absolutely. The voiding of the material, while we absorb much of the food we ingest, we do not absorb all of it. So defecation is the removal of all unabsorbed. Oops, there. Plus other stuff out of the body. And if we have to get things out of the alimentary canal, what else do we have to do?
Bring it in.
Bring it in. What would we call that process? Stuff in our face? There you go. I like ingestion better. That sounds better than stuff in our face. Excellent. Perfect. That's 5. I can think of at least one more. All of these processes, chemical digestion, mechanical digestion, absorption, defecation, ingestion, do all of those occur in the exact same location?
No.
No. So what else do we need to do to this food? There we go. Amy's got it. Jane got it too. We need to move it from one place to another. Propulsion. We need to move that food through the alimentary canal. Excellent. Perfect. So those are our 6 big basic functions of the alimentary canal. And I've written it out here, but we also have the pretty words here that kind of show that process as well. Now, there is one key that I want to emphasize about this. Notice I have this as a list of 1 through 6. However, is this truly steps of a process? I mean, clearly some of these things have to happen before others. Obviously, ingestion has to occur before absorption can take place. But do we, after we ingest, do we then do all the propulsion and then all of the mechanical digestion and then all of the chemical digestion and then all of the absorption and then all of the defecation?
No.
No. Every organ in the alimentary canal does one or more of these functions. And conversely, most of these functions occur in multiple locations. Again, I do say most because like ingestion obviously just occurs in the mouth. Defecation just occurs from the rectum and the anus. So again, obviously there are some of these processes that do only occur in one location. But the other 4, propulsion, mechanical, chemical, digestion and absorption. Those can all occur in multiple organs. And your book actually has a nice picture that emphasizes this point. So here we see, and again, in the words, the point I'm trying to make, and then also this simple illustration from your textbook that kind of emphasizes all the locations where all these things are occurring. Obviously, propulsion has to occur throughout the entire digestive system, but absorption occurs in multiple locations, chemical digestion occurs in multiple locations, mechanical digestion occurs in multiple locations. And so that's one of the things that we will emphasize as we are working our way through this organ system. All righty. Questions on that? All right, excellent. All right. Before we start our mystical, magical journey through the alimentary canal, there are some basic concepts, basic functions we need to understand about how the digestive system works. As I mentioned, it's somewhat unique in that all of the processes that take place within the digestive system basically occur outside the body. So we are regulating and modifying the environment outside of the body, which is somewhat unique. Again, because that lumen is the topological exterior of the body. And while we talked about those 6 main functions of the digestive system, there's basically 2 keys to our digestive process. Right? There is going to be some type of stimulus. Many of these, in fact, I'll write this up here. Most processes occur via reflex arcs. Is that something we voluntarily control?
No.
No, absolutely. So we need some type of stimulus, typically some type of mechanical or chemical stimulus, like a stretch of the stomach or a change in the pH or the presence of a particular amino acid or fatty acid or something along those lines. And from that stimulus, basically one of 2 things are going to occur in our alimentary canal. We are either going to change the glandular activity, increasing the amount of something we secrete or decreasing the amount of something we secrete, or we're going to change the motility of the organ, change the speed of its contractions, or change the order of the contractions or the strength of the contractions. We're going to somehow modify either the glandular activity or modify the smooth muscle activity. Now, as I mentioned, these are primarily reflex arcs. These are, and certainly, let's do it this way, involuntary processes. So there are going to be 2 main ways we are going to control or regulate digestion. One of them clearly is the nervous system, because it says it here. What would the other one be?
Endocrine?
Yeah, hormones, endocrine system, absolutely. So there are going to be 2 main regulatory processes for the nervous system. I mean, for the digestive system, nervous and hormonal. Let's talk about the nervous mechanisms, plural, first. The first is actually what we call an intrinsic system. Intrinsic, of course, means it is contained within. There is an elaborate network of nerves in our digestive system known as the enteric plexus. And this enteric plexus is often affectionately known as the gut brain. because of a lot of the decisions that need to take place in the digestive system. Those decisions are made right there in the digestive system. Let's talk about the most basic example. You have that cheeseburger for breakfast. And when you have that cheeseburger for breakfast, as it goes down your gullet into your stomach stretches because you've put food inside of it. All right. Putting that food inside of your stomach causes the stomach to stretch. There are stretch receptors in the wall of the stomach that send a signal to this enteric plexus, not to our brain, not to our spinal cord, but to the stomach itself. This elaborate network of nerves in the stomach. And when it stretches, the stomach says, I've got food inside of me. I need to start churning that food up. This intrinsic control is basically what we call these short reflexes. Again, it's controlled locally by the enteric plexus. And it's stimulated by a local cue, like the stretch of the stomach. And its effect is going to be local as well. Let's switch to the whiteboard and talk about this. We are talking about our neural control of the digestive tract. And basically there are two reflexes. The first is our short reflex. And so guess what the other one is?
Long reflex.
There you go. Or we can also think of them as being intrinsic control and an extrinsic control. Actually, let's put it up here. So we have some organ like the stomach. With that intrinsic control, as I mentioned, again, it's a reflex, so we need a stimulus. The stimulus here with our short reflex comes from inside the organ. All right, because we know all reflexes have to have some type of stimulus. It comes from inside the organ. And as I said, this can be something like the stretch. This could be a change in the pH. This could be some type of certain, like I said, substance, like a particular amino acid, or the presence of fatty acids, or the presence of glucose, or something like that. Some type of substance or something like that that is going to be the stimulus. And of course, we need some type of local receptor. Right? Because again, like I said, my wife this whole time has been sitting next to me off camera, blowing a dog whistle. Right? Has it changed anyone's behavior here in the classroom?
No,
No, why not?
because the hertzes are too big for our ear.
Yeah, exactly. None of us are dogs. None of us can hear a dog whistle, so it wouldn't change our behavior at all. So obviously we have to have some type of receptor that is capable of perceiving that information. And so here it's going to be something like a stretch receptor or a chemoreceptor, or even an osmolarity receptor, something like that, some type of local receptor inside the organ that is perceiving that information. Now, of course, as we know, we need in our reflex a command center, some place to process the information. And as I mentioned, with our short reflex, that processing of the information occurs within that enteric plexus. And again, this enteric plexus is basically just an elaborate network of nerves and ganglia in the organ. Let's say it this way, in the organ's walls. So within the walls of that stomach is this elaborate network of nerves that processes the information. And of course, once it processes that information, it then makes a decision. And again, are these going to be big decisions like deciding, right, what college you're going to go to, what do you want to be when you grow up, who you're going to get married to? These are the kind of decisions your enteric plexus is going to make.
No.
No, of course not. So basically, we are going to have some type of effect, and that effect is typically local. Of course, that means there can be some wider spread effects, but it's typically going to be local. And as we mentioned, it's going to either cause a change in secretion and or motility of the organ. So it's all local. So again, we get that cheeseburger into our stomach. That causes the stomach to stretch. There are stretch receptors in the wall of the stomach, sends that information to the enteric plexus that is located within the stomach. And actually, let's write that out. That enteric plexus in the stomach, which makes a decision and then tells the stomach what's to do. And as a result of that, the stomach increases motility, starts churning up the food, or it increases its glandular secretions. It makes more acid. We comfortable with that? Again, it's a reflex. It isn't something that we haven't talked about before. We have a stimulus, we have a receptor, we have a command center, we have an effector. We have all those things we expect from a reflex. So questions on that?
Professor, can I ask a quick question?
Absolutely.
Off topic, but my uncle got stomach surgery. He removes like the big portion of his stomach. The purpose is to lose weight.
Mhm.
And the doctor explained not only he removed the large portion of the stomach, but he also removed the nerve, and he called it stomach brain. That basically he explained that he does not get hungry or something. So is that the enteric plexus that has been removed, you think?
There isn't, it
didn't happen.
Okay.
So
Well, no, okay, so here's my hesitation. As I've talked about in this class, often I use the phrase, the sky is blue, to imply that I'm not necessarily lying to you about this information. I'm just simplifying it a little bit so that it makes a little bit more sense, because we don't need to go into the deep depths of information that someone who is studying this for, their career would necessarily go into. Clearly, that was what he was doing in this case, and I'm trying to understand what it was that he was trying to tell him. The problem with that piece of information is there is no nerve that tells us when we're hungry, right? That's not what In fact, we talked about that a little bit when we were talking about the hormone leptin, right? Leptin is a hormone that's supposed to make us feel full so that theoretically we would stop eating, but most of us continue to eat as long as there's food on our plate, right? Or if we're distracted by a movie, we'll continue to eat whether we're full or not, or if we're stressed, or if we're angry, or if we're bored. I mean, there's all sorts of other reasons. So even things like that that could directly impact it aren't very effective. So even if there was a nerve that made you hungry, it wouldn't probably be that effective, or affected your hunger, it wouldn't be that effective. And more importantly, there isn't. Now, in removing the parts, he may have removed some of the neural controls, which we'll talk about in just a second here with the long reflex that relates to what we're talking to, that could decrease the activity of the stomach. And if it decreased the activity of the stomach would have a harder time emptying. And if the stomach was having a harder time emptying, if it got full because there's less of it, and it stayed full for longer because it wasn't getting that neural input to stimulate it, then that could give you a sensation of feeling full and not wanting to eat more that way. So my guess is that's probably what he was talking about.
Yes.
That's not that he would not be hungry anymore, but that he would get, because of a smaller stomach that was taking longer to empty, he would probably feel full sooner while he was eating, right? Because again, it's like when you eat at Thanksgiving and you overfill your stomach and it gets uncomfortable and you don't want to eat anymore, right? If you shrink the stomach and you make the stomach stay fuller for longer, then you're going to get that sensation sooner. So my guess is that was probably what he was referring to. Does that make sense?
Yes.
Okay, excellent.
Yeah.
All right, which is a perfect lead in, actually. I know you said it was off topic, but it really wasn't. A perfect lead into what we're going to talk about next, this long reflex. It is still the reflex, so we're still going to have all of the same basic components. We are still going to have a stimulus. All right, so let's make this the right size and the right color. Now, as I mentioned, you eat that cheeseburger hits your stomach, it stretches your stomach out, and that affects your stomach activity. But do you actually have to eat the hamburger to stimulate your digestive activity of your stomach? What if you're just standing next to the barbecue while it's being cooked, and you hear the sizzle, or you smell that Maillard reaction as it browns and gets all crispy and all of that, or even see it? Heck, you don't even have to smell it or anything else, right? There is nothing in the world that looks better than a Carl's Jr. hamburger on their commercials, right? In real life, they don't look anything like that, but a Carl's Jr. hamburger on their commercials are the best looking things ever when it comes to food. And so just sitting there staring at one of those cheeseburgers, or hearing the sizzle or smelling it, can that stimulate your activity of your digestive system? Yeah, absolutely. Absolutely. So here, in the case of our long stimulus, the stimulus can actually come from inside the alimentary canal, or even outside of it, right? Like vision, like smell, like that, and even thinking. If you really were to sit in a room for 5 minutes with your eyes closed and think about that amazing meal that you had with your significant other at your last anniversary, where you locked up the kids in the basement, you got some free time, you put the cat in charge of the house, you went out to a fancy restaurant, you put on pants, right? Again, I guess this was probably a couple of years ago now because of the whole COVID thing. But if you really sat there and really thought about that meal and the different courses and how that melted butter was, how that herb butter was melting on the top of the steak, if you really sat there and thought about that, do you think you could get yourself to salivate just from thinking about it, not even seeing or smelling or hearing the food?
Yeah.
100%.
Yeah, absolutely. So one of the keys with this long reflex is it can actually be top down. It can actually be neural. Just thinking about these things can cause that stimulus. So in this case, the stimulus can be inside the organ, but it can also come from outside of the organ. Excellent. Obviously, when we're dealing with this, then we are no longer dealing with local receptors. As I mentioned, these receptors can be our special senses, like vision, like smelling, like hearing, like tasting. But as I also mentioned, it can also be neural, it can be cognitive, where we're just thinking about it. This is often referred to as top-down processing, where we're thinking about something and that has an ability to affect our body. So it's not necessarily just a local receptor. There are many receptors that could be capable of this. Yes, it can be local, but it could also be from outside, like I said, have special senses, or even just the thinking about it. Now, one of the big keys with the long reflex, obviously besides of the location of the stimulus, is where the information is processed. In this case, the information is processed in the central nervous system. Now, again, when I say the central nervous system, is it you saw that cheeseburger on that Carl's Jr. commercial and you said, hey, stomach, that looks really yummy. You should start gurgling and producing hydrochloric acid?
No.
No. So we're going to, when we talk about these digestive functions, we're talking about in the medulla oblongata or in the hypothalamus even. Right, somewhere in the brainstem. So again, this is still unconscious, subconscious, not voluntary. This is still involuntary processing that is taking place, but it is occurring in our central nervous system. And then our central nervous system, through pathways that we will talk about, then send the information to the enteric plexus to modify its behavior. So in this case, we are starting out here in the central nervous system. Right. Again, this central nervous system is getting the information from the organ, or it's getting the information from outside the organ, processing that information. And once it processes that information, it then sends a signal to the enteric plexus. And once again, the enteric plexus then tells the stomach to change the motility, change the glandular secretion. And again, I keep talking about that example of seeing that Carl's Jr. cheeseburger on the commercial and you salivate and your stomach gurgles in response to that. But if you're a vegetarian, are you going to salivate and is your stomach going to gurgle in response to seeing that cheeseburger? Or smelling that tri-tip on the barbecue? If you're vegetarian, are you going to have that kind of a positive increase in activity as a result of that?
No.
Again,
Probably not.
these are the easy questions, folks. Excellent. No, of course not. So obviously, again, when we're dealing with these outside controls, this long reflex, again, we are dealing with these cognitive effects. So smelling, burnt meat on the barbecue doesn't have the same effect on everyone because of these differences of the cognitive, right? Maybe you grew up with a cow as a pet, and so you can't eat beef as a result of that, right? Or like I said, or you're vegetarian because you spent a summer on a farm, or maybe you don't eat it because of religious or cultural significance. Whatever it is, notice there's a cognitive component where not everybody's going to salivate as a result of it. Right? It's different with the short reflexes. If I held all of you down and I shoved a piece of meat in your mouth and I forced you to swallow it, once it got to your stomach, does your stomach care that it's meat and you don't like to eat meat?
No, it's still gonna work the same way.
Yeah, it's still going to break up that food, right? You'll be cognitively upset by it, probably emotionally upset by me doing that to you, but the stomach doesn't care. The stomach stretches, the stomach responds by doing its activity. But notice with these long reflexes, we can, because of that cognitive component, it can have a different effect. And here's the other big key to our long reflex as well. With that effect, it can be both in the organ of the stimulation and in other organs as well. Say it that way. I like that better. So it's, instead of a local effect, it can have a broad effect. Like I said, tasting that meat, assuming it is something that you enjoy, will make your salivary glands become more active in your mouth, but it will also increase the activity of the stomach as well. So when our central nervous system is influencing things, it will also influence other organs outside of where the stimulus was as well. Again, simple illustration, but hopefully it emphasizes the point. Again, notice as we talked about with that intrinsic control, that short reflex, the stimuli is occurring inside the organ of the alimentary canal. It's being processed locally in the enteric plexus, and its effect is local. Extrinsic controls, our stimulus can be either inside or outside of the alimentary canal. It is going to be processed by our autonomic nervous system centers in our primarily medulla oblongata and also the hypothalamus. And we'll go ahead and do this now. We, of course, have two branches of the autonomic nervous system. What are the two branches of the autonomic nervous system?
Parasympathetic and sympathetic.
We won't worry about the pathways too much right this second, but anybody know the name of the nerve or nerves that control the digestive system in the parasympathetic pathway?
Vagus nerve.
Yeah, there you go. Cranial nerve 10, vagus nerve. Gave you the easy one to start. Anybody know what nerves, what set of nerves control the digestive system, specifically the organs below the diaphragm? There was a special name we had for that bundle of nerves that control the organs below the diaphragm. Anybody remember? It's time to start remembering because we will talk about them a lot. The splanchnic nerves. So if you remember, the splanchnic nerves are the ones that come off of the three collateral ganglia, the celiac, superior mesenteric, and inferior mesenteric ganglia, and they innervate the organs below the diaphragm. So it is the splanchnic nerves for the sympathetic nervous system and the vagus nerve for the parasympathetic nervous system. And in general, what kind of an effect does the parasympathetic nervous system have on digestion?
Excitatory.
Yeah, typically it's going to be excitatory. And our sympathetic then, of course, is going to be typically inhibitory. Excellent. And much like I did on my simple illustration, your book's got a nice illustration that kind of shows this as well. Notice for the short reflex, we have a stimulus in the organ. processed by receptors in the organ. Information is sent to that enteric plexus to make a decision locally, and it has a local effect on the organ. Our long reflex can start with a stimulus inside the alimentary canal or a stimulus outside. Here, the information is processed in our central nervous system and then influences the enteric plexus to affect some change. But notice what it's lacking is a reminder that can also affect other organs outside of the organ where the stimulus was as well. So another nice basic illustration that shows these two main types of neural control. All right. Questions on that? All righty, excellent. Like I said, we don't just rely on neural control. There is also hormonal mechanisms as well. Rather than sitting here and rattling off all of the hormones that you're going to be responsible for, I'll just remind you right now that the digestive system, like we know, includes things like the pancreas that we know have endocrine functions, producing insulin and glucagon and things along those lines. But most of the organs of the digestive system have the ability to produce some hormones. Within their mucous membranes, they have special cells called enteroendocrine cells that are basically, it's just a fancy way of saying, a single-celled endocrine gland. So these enteroendocrine cells are single cells that are capable of producing and releasing hormones. And there are a lot. There are almost 20 hormones that are produced and control the function of the digestive system. We will not talk about all of them. Your book does a good job of mentioning most of them. And we'll talk about a select few of them that I think are important and that we will emphasize. And again, not being hormones, not surprisingly, they have the ability to not only affect the organs of the alimentary canal, but also, because these are chemical signals that get in the blood and can go anywhere, they have the ability to affect our accessory glands as well, like the liver, like the gallbladder, like things along those lines. And of course, our salivary glands and things along those lines as well. And again, being hormones, they can even influence structures outside of the digestive system as well. All right. Questions on that? All right. Now that we have a basic understanding of the function, we are going, and actually I'm going to cheat and I'm going to stay here for a second. We are going to talk about the basic anatomy of the alimentary canal. Again, it's that 30-foot tube from mouth to anus. Now, as we have known, because we've talked about it, the alimentary canal in the oral cavity, in the pharynx, in the proximal esophagus, is all highly specialized. One of the ways, for instance, it's specialized is the epithelial tissue you find lining that region. And what is the epithelial tissue that lines these particular organs of the alimentary canal?
Areolar.
Sure, that is the connective tissue, but what's the epithelial tissue?
Non-keratinized stratified squamous.
There you go, a non-keratinized stratified squamous epithelial tissue. I'm just going to abbreviate it here because it's not super important for the moment. Absolutely, because that's the part that is open to the outside world. That is the part that needs to be protected from hot coffee, spicy tortilla chips, right? Sharp tortilla chips, things along those lines. And as we also know, the distal part of the digestive system, primarily the rectum and the anus, have that same non-keratinized stratified squamous epithelial tissue and other specializations to it as well. So as we work our way through the organs of the alimentary canal, we will talk about some of the specializations of that. But your rectum and anus aren't that big. Your mouth, pharynx, and esophagus aren't that big. So the vast majority of your alimentary canal basically falls in between. And what's nice about this is basically we're talking about the distal part of the esophagus, the stomach, all of the small intestine, and most of the large intestine all have the same basic anatomy. And that's what we see here. When we talk about the basic anatomy of the alimentary canal, we're not talking about the two specialized ends. We're basically talking about everything in between. So again, we're talking about the distal third of the esophagus, the stomach, all of the small intestine, and most of the large intestine. So that is another right. There we go. All of it have the same basic anatomy, and that is what we see here. So what we're going to do, and this is a good breaking point. It's a teeny bit early, but it's still a good breaking point. When we come back is we are going to identify and discuss the 4 main layers and all of the sublayers and all of the other additional structures found in the basic anatomy of the alimentary canal. So we'll take a look at this, we'll draw it, we'll talk about it, make sure that we understand it. All right, questions on that? Or any of the material we just covered? All right, let's go ahead and take our first break. We'll take a 15-minute break. It's 1 o'clock now, so we will restart at 1:15, and I will start the recording at that time. All right. If there are no questions, then I will see you in 15 minutes. All righty, let's get started. As I mentioned, our goal here is to identify the basic anatomy of the alimentary canal. And as I mentioned for this, there are 4 specific layers. So let's do this from the, we'll go from the lumen. Oops, hold on. Oh, too late. So here is the lumen of the alimentary canal towards the center here. And the first layer is what is known as the mucosa. Or, of course, the mucous membrane. Both of those are acceptable. And being the mucosa, we should know that it is comprised of an epithelial tissue sitting on top of a connective tissue. So what is the epithelial tissue that lines the mucous membrane of the majority of our alimentary canal? Now remember, it's non-keratinized in the oral cavity and the anus and the rectum, but through like the stomach, through the small intestine, what is the epithelial tissue we found there?
Simple squamous epithelial tissue.
Close. It is simple, but not squamous. Simple columnar. There we go. Excellent. So this is a simple columnar epithelial tissue, which of course, as we know, has tall, narrow columns, easy to recognize and identify. Again, one of the characteristics that is distinct about it, helps to make it easy for us to be able to recognize, is the nuclei tend to all line up fairly uniform. So they're fairly uniform in size, fairly uniform in shape in the organization. And what do many, not all, but many of these cells have on their apical surface?
Villi.
Careful, microvilli. Right, they're going to have microvilli. Villi are something else we'll talk about entirely. But you are correct, it is going to have the organelle, microvilli, on their apical surface. Many, not all, but many. So let's say that. That microvilli on the surface. Excellent. And of course, what is the function of the microvilli?
Increase the surface area.
Yeah, increase the surface area for absorption of material or for chemical digestion or for things along those lines. Excellent. And what type of connective tissue is our simple columnar epithelial tissue sitting on top of? What type of connective tissue always helps to form a mucous membrane? Excellent. Areolar connective tissue, spectacular. However, we know something else about this areolar connective tissue as well. An areolar connective tissue that is part of a mucous membrane. Remember, we give it a name based on its location. And what do we call an areolar connective tissue that is part of a mucous membrane based on its location? I know this is some muscle stretching. You haven't thought about membranes since we did epithelial tissues and connective tissues in 430. But what did we call an areolar connective tissue that was part of a mucous membrane?
Was it the mesothelium?
Close. Mesothelium, remember, is the simple squamous on a serous membrane. So it is definitely a locational name. So you have the right idea. You're on the right path. But for our areolar connective tissue, it was a lamina propria. So it's an areolar connective tissue that, based on its location, we call a lamina propria. So again, you may not recall, but this is definitely something that we talked about back in 430. And that is definitely something that is seen here in our mucous membrane of our alimentary canal of our stomach. However, there is actually a third sublayer to our mucosa in the alimentary canal. There is indeed that simple columnar epithelial tissue. There is indeed that areolar connective tissue that we call a lamina propria. But underneath that is a third layer. And this third layer is actually a small thin layer of, and since it's a thin layer, we'll go with that color, of smooth muscle. So we have this thin layer of smooth muscle that is known as the muscularis mucosa. Muscularis because it is a muscle layer of the mucosa. Now, why might we want a thin layer of smooth muscle at the base of our mucous membrane in our alimentary canal?
For motility?
Yeah, we can change the shape of it, which again can increase and decrease surface area. right? It can dislodge, right? You get that little bit of peanut butter stuck on the roof of your mouth and you need to use your tongue to wiggle it off of there. So if a little chunk of the food gets stuck on the wall, we can help to wiggle it off that way. So we can change the surface area. We can gives it a little bit of motility so we can move and shift. Again, these aren't going to produce powerful enough contractions to move the entire, bolus of food through our stomach, for instance, but can it cause minor changes in shape of the small intestine or something like that that could affect surface area or could affect delay movement or dislodge something that's stuck? Yeah, absolutely. All right. So those are our 3 sublayers of the layer closest to the lumen, that is the mucosa. Now, deep to the mucosa is a layer known as the submucosa. This layer, the submucosa, is comprised of areolar connective tissue. Is this a lamina propria?
No.
No. Now, is there anything different between this areolar connective tissue and this areolar connective tissue? The tissue itself.
No.
No, it's areolar connective tissue, right? Areolar connective tissue is a loose connective tissue, and there's no difference other than their location. This one is not a part of the mucosa. It is not a lamina propria. It's on the deep surface of the muscularis mucosa, away from it. And typically a larger layer than what we see in the mucosa itself. And as we know, areolar connective tissues are loose connective tissues. Let's get rid of those stars because we don't need them. A loose connective tissue, which means there's room for lots of stuff. And that's what we have in this space. This submucosa contains a lot of stuff. One thing that will be contained inside here are blood vessel capillaries. Why might we have blood vessel capillaries inside of this space?
To absorb the nutrients.
Yeah, absorb nutrients. Lymphatic capillaries. Why might we have those in this space?
For fats like the lacteals.
Yeah, the lacteals would be an example of one of those lymphatic capillaries. It doesn't have to be a lacteal. Lacteals, as we see, as we'll see, are going to be unique to the small intestine. But yeah, lymphatic capillaries are going to be able to help us in the absorption of fats or fat-soluble vitamins or things along those lines. Glands for our secretion, so large glands that are going to be here for the secretion to release of that material. And the other thing that's going to be in here is one of the parts of the enteric plexus. Remember, the enteric plexus is responsible for 2 things: controlling smooth muscle and controlling glands. Well, the part that is found here in the enteric plexus is called the submucosal plexus, because they're not very original. And what of those 2 functions do you think it's going to be responsible for? Is this going to be responsible for controlling the smooth muscle, or might it be responsible for controlling those glands that are here in the submucosal area? Not a trick question.
Glands.
Yeah, there you go. Excellent. So it's responsible for controlling the glands. So there's all this hunk and junk within this submucosal layer. All right. Questions on that? All right. Our next layer is what is known as the muscularis externa. Although I will say sometimes it is just simply called the muscularis. It would be fine to call it the muscularis. But muscularis externa helps us distinguish it from muscularis mucosa. And guess what it is comprised of? Again, not a trick question. What do you think the muscularis externa is comprised of?
Muscle.
Excellent. And what kind of muscle do you think?
Smooth.
Smooth muscle. There you go. Sorry, Catherine, I didn't see you right there. This is where we have the smooth muscle layers, plural. One of those layers, the one closest to the lumen. Oops, nope. Is a circular layer of smooth muscle. So this one goes around the circumference of the organ. And what do you think the circular muscle would control? Well, if you have a circular muscle and you around the circle of a lumen and you contract that muscle, what happens?
Closes the circle.
Yeah, the circle gets smaller. If you relax the muscle, what happens to the diameter of the lumen?
Increases.
Yeah, so this controls the diameter of the lumen. In some portions, this can become enlarged. And if it becomes enlarged, not only can it decrease the size of the lumen, but it could actually close the lumen, acting as a valve. And what would we call this thickened circular layer that controls the movement of things through it? There we go. Emmy's got a sphincter. Excellent. But there is a second layer of smooth muscle as well. This second layer of smooth muscle is a longitudinal layer. Longitudinal layer goes the entire length of the organ. And so what do you think a longitudinal layer would control?
The length of the alimentary canal.
Absolutely. If we contract the longitudinal layer, we get a shortening of the organ. If we relax it, we get a lengthening of it. So it controls the length of the organ. Absolutely. Perfect. Not surprisingly, we have a second portion of elaborate nerves found in this area. Another part of the enteric plexus. And what do you think this part of the enteric plexus does? Come on, guys. These are the easy answers.
Does that help it glide easier between each other?
Well, again, these are nerves that are embedded between the two layers of smooth muscle. What do you think these nerves embedded between the two layers of smooth muscle control?
The smooth muscle?
Yeah, they control the smooth muscle, exactly. And this one is called muscularis plexus. Excellent. All righty. That brings us to our last layer, the outermost layer, furthest from the lumen. And this can be one of two things. It is going to be either an adventatia or the other thing that it is going to be is a serosa. Oops, hold on. So let's draw a line down the middle here. And let's talk first about the one we're probably most familiar with, and that is a serosa, which of course is a fancy way of saying what?
Serous membrane.
Yeah, right. Serosa is just the noun, whereas serous is the adjective that describes the noun. And we can be more specific here as well. While serous membrane is the general term, serosa is the general term, how would we call this serous membrane that was lining the outer surface of an organ?
Was it the visceral peritoneum?
That is exactly what it is. Excellent. So this is our visceral peritoneum. And luckily, we know that serous membrane is again going to be comprised of a connective tissue sitting on top of sitting underneath a superficial epithelial tissue. And so what would the epithelial tissue be from a serous membrane? This we can steal right back from when we were using the cardiac muscle, talking about the anatomy of the heart. What was the epithelial tissue that forms a serous membrane?
Simple squamous.
Simple squamous, excellent. And as someone mentioned earlier, based on its location, what do we call a simple squamous epithelial tissue that is part of a serous membrane?
Mesothelium.
Mesothelium, excellent. Excellent. Jacob, I think, mentioned that earlier. And of course, what connective tissue is it sitting on top of?
Areolar.
And we can even take it one more step further than that. What do we know that a serous membrane produces?
Serosal fluid.
Yeah. And again, is the goal to make as much serous fluid as possible?
No.
No, we produce a thin layer of serous fluid. And when we produce that thin layer of serous fluid, what do we call that based on its location?
Transudate.
There we go. Excellent. So let's cheat a little bit. We can move this down and out of the way. Move this down a little bit, and then that would give us our little bit of room here to draw a little bit of an areolar connective tissue sitting on top of a simple squamous epithelial tissue. We'll just make 1, 2 flat cells with a thin layer, a thin layer of that transudate sitting on top of it. Excellent. However, as I mentioned, not all of the organs in the alimentary canal are going to have a visceral peritoneum on them. So those that do not have a visceral peritoneum, or let's say it this way, the ones that do have a visceral peritoneum, these are the ones that are actually located within the abdominal pelvic cavity. Well, so where the heck are the other ones? Well, typically the other ones, those that are lined with an adventatia, are not within the peritoneum, let's say it this way. Instead, they are typically anchored to the walls of the abdominal pelvic cavity. Often posteriorly. And because they're anchored in the walls posteriorly, they aren't going to move around. And if they're not going to move around, they don't need that slippery surface. So basically, instead, what we see here on the outer surface is basically just a fibrous connective tissue outer surface. So we have a dense irregular connective tissue. And its job, like I said, is to basically anchor the organ in place. All righty. So you either have an adventatia or a serosa. You can't have both. I've taken the time to do this, but this shouldn't be a new concept for you, right? If you think about way back in 430, when we were talking about membranes, we talked about how you would build an organ. Let's say, for instance, this is the abdominal pelvic cavity. And so you have the esophagus coming down into the abdominal pelvic cavity. And then in the center of the abdominal pelvic cavity, you have the stomach, which connects to the small intestine, and from there goes to the large intestine and everywhere else until it's finally out of the alimentary canal. And we can start labeling things that we know. As we mentioned, the wall of our abdominal pelvic cavity is going to be lined with a parietal peritoneum. Might be a little too small. We know the organs are going to be lined with a, and I'll put it over here, visceral peritoneum. And we know on the inside, organ, because our digestive system is open to the outside world, is lined with a mucous membrane. So if you think about that doctor who, when you swallowed that penny, let's say it got stuck in your stomach and they had to open up your stomach to get to it. If you think about the layers they would have to cut through to get into it, Well, first on the outer surface here, we would have that visceral peritoneum, which is a serous membrane. And of course, as we know, that serous membrane is going to be made up of a areolar connective tissue. And on top of that is going to be a simple squamous epithelial tissue. And on top of that is going to be our thin layer of transudate. So again, we have a little bit of areolar connective tissue, a single layer of flat cells, and then that little bit of transudate on the outer surface. On the inside, we know we have a mucous membrane, and apparently I spelled mucus wrong, but as we know, is going to be a simple columnar epithelial tissue sitting on top of an areolar connective tissue. And that would be here on the inside. With our microvilli and a little bit of areolar connective tissue. And at the time when we were talking, we mentioned then you would just have smooth muscle in between. We now know it's a little more sophisticated than this. We know we are going to have 2 layers of smooth muscle. Closest to the lumen is going to be circular. Furthest from the lumen is longitudinal. Smooth muscle. But we also know that there's a little bit more in between this as well. So I got to cheat, sneak that down. There. And there. Because now we know in between there is also going to be a submucosa. And we also know that there is a 3rd layer to this mucous membrane, and that 3rd layer to the mucous membrane is that muscularis mucosa. So a thin layer of smooth muscle in here as well. And again, as amazing as my drawings are, the easiest place to see this is actually the pretty picture from your textbook. So remember, great question. Sorry, I didn't catch that one before. Let's go back to the picture. Remember the epithelial tissues. Well, actually, let's do it this way. Epithelial tissues are always superficial. They face the open spaces. So on the inside of the stomach, we would have a connective tissue that would be deep, and the epithelial tissue would be on the surface, the inner surface, lining the lumen of the alimentary canal. On the outside, we would have the connective tissue, and it would be deep. And in this case, the epithelial tissue would be superficial, facing this cavity, the space that is the parietal peritoneum. So all the other hunk and junk is in between the two. But notice basically they're opposite of each other because we always need the epithelial tissues to have a free space above them. So whether that free space is inside of the abdominal pelvic cavity or that free space is inside the lumen of the stomach, the connective tissues are going to be away from the surfaces and the epithelial tissues are going to be towards the surfaces. Does that make sense?
Yes, yes, thank you.
Excellent. Perfect. And like I said, part of it could easily be my drawing of these things. So let's actually take a minute and actually look at this with the really pretty picture that an actual artist drew for our textbook. So again, notice if we started from the lumen, here is the inside, here is where the cheeseburger sits. Notice we have our simple columnar epithelial tissue. I don't like that. Our simple columnar epithelial tissue lining the surface. Deep to that is our areolar connective tissue that, as part of a mucous membrane, we call the lamina propria. Notice also in that lamina propria, we happen to see your friend and mine, our mults. What's a mult again?
Mucosa associated lymphoid tissue.
Yeah, basically it's a follicle. We have a lymphatic follicle there with its germinal center, with its lymphocytes, providing that protection. And notice some minor glands and things along those lines. But notice we also have a third layer to our mucosa, and that is that thin layer of smooth muscle that is the muscularis mucosa. And actually, let's go through this way. So our epithelial tissue, simple columnar, our areolar connective tissue, the lamina propria, and the muscularis mucosa. These three layers all help to form the mucosa of our alimentary canal. It's important to distinguish this because, again, as we know, deep to this is the submucosa. The submucosa is also areolar connective tissue, but this is where we find the large glands. This is where we find that submucosal nerve plexus. This is where we find large blood vessels that will be absorbing from the capillaries and absorbing the materials. Our lymphatic capillaries, where we're absorbing that material. Next, we have the two layers of smooth muscle, circular, notice going around the entire circumference of the organ, longitudinal running the length of it. And in between, we have that myenteric nerve plexus that controls the smooth muscle. And oops, like I said, here on the outside, we see an example of a serosa, a simple squamous epithelial tissue with a little bit of an areolar connective tissue. And notice we see one other thing as well. We're going to talk about this muscularis more in a second, but I want to, because we were talking about these layers, come back to this and do one other thing. Here's my amazing drawing of the alimentary canal from a coronal section. But what if we did a transverse section as well? Here would be, of course, your abdominal pelvic cavity. And here, of course, oops, stop it, would be your stomach. As we know, that visceral peritoneum is going to line the outer surface of our stomach. And our our parietal peritoneum is going to line the walls of the abdominal pelvic cavity. Because as we know, serous membranes are double membranes. But if you remember, and we learned this, or at least we learned it in 430, we were reminded of this again in 431 with the heart. Remember, the visceral and the parietal serous membranes connect to each other. That's a little bit more tricky when you have an organ like the stomach that sits in the middle of the abdominal pelvic cavity. So what actually has to happen is that our parietal peritoneum actually has to extend off the wall to attach to the visceral peritoneum. And then again, it has to extend off to come back. Notice what this produces is a double layer of serous membrane that basically anchors the organ loosely in place. This double layer of serous membrane also provides a passageway for blood vessels and nerves. So our blood vessels, our arteries, our veins, our nerves are all able to get to and from that organ. But essentially, this is just a serous membrane. So as we know, it is a single layer of flat cells, a simple squamous epithelial tissue with a little bit of areolar connective tissue underneath, and then a little bit more. So this areolar connective tissue holds those nerves and blood vessels in place, and it's just held here by sandwiched in between two simple squamous epithelial tissues. And if you really scratch your head and think back, we actually came up for a fancy name. What is the fancy generic name for this double layer of serous membrane that anchors this organ in place? Does anybody remember? Starts with mesentery.
Mesenteric canal?
No, just mesentery. Exactly. Excellent. It's the mesenteries. So this mesentery comes off. I mention this because if you look back at our picture, lo and behold, here we see a simple squamous epithelial tissue, a second simple squamous epithelial tissue with the areolar connective tissue holding our blood vessels, our nerves. Oh, I even forgot about our lymphatic vessels going in there. And lo and behold, that's our mesentery. So that adventitia, pardon me, not the adventitia, the serous membrane forms the mesentery. That serosa forms the mesentery. So here we see those layers nicely. Now I want to talk a little bit more about the smooth muscle layers, because as we talked about, the advantage of having two different orientations to these muscles is they have two different effects. And those two different effects can allow for differences in function. One of the simplest example of this is where we get rhythmic alternating contractions of the two layers to produce a process called peristalsis. And peristalsis is primarily for propulsion. Basically, what peristalsis is we squeeze the tube and then we shorten, and we squeeze and we shorten, and we squeeze and we shorten, and we squeeze and we shorten. And as we make those alternating contractions, and I think I have a better picture of that here, these alternating contractions of the circular and then the longitudinal and then the circular and longitudinal, we can move the food from one place in our alimentary canal to the next. However, our smooth muscle can be used for other functions as well. Notice here is a muscle activity known as segmentation. Segmentation occurs only in the small intestine. And notice with segmentation, what happens is you get an alternating contraction of the circular layer. So first this part of the circular layer contracts, and then that one, and then this one, and then that one. Back and forth and back and forth and back and forth. And notice as it's going back and forth, is it doing a really good job of moving the food from one part of the organ to the next?
No.
No, it's pretty much just mashing it back and forth. So this serves two important functions. This helps with our mechanical digestion. I know this is going to be shocking to many of you because I know everybody here in this class chews their food, takes a bite of food and chews it the 24 times you're supposed to chew it before you swallow it down. Right? So I know everybody in here does that, but believe it or not, some people will actually take a bite of food, chew it twice, and then down the gullet it goes. Right? I know, shocking information. So having this mechanical activity can help to continue the mechanical breakdown of the food. It also basically is rubbing the food back and forth across the surface of the small intestine. That surface of the small intestine is lined with enzymes. So this also helps in our chemical digestion by rubbing it across those enzymes again and again. And it's also rubbing it across the microvilli. So this also helps in absorption as well. Because the more the food is in contact with the small intestine, the more absorption can take place. So notice we're still using the same muscles. We're just contracting them in a different fashion and getting a very, very different function from them. And there are other examples as well. We won't go into tremendous depth about it, but like for instance, in the large intestine, we can have mass movements and haustral contractions and other functions we'll talk about when we get to those specific organs. All right, lastly, as I mentioned, you either have a serosa, which is the visceral peritoneum and forms the mesenteries, or an organ may instead form an adventitia. Now, I want to talk a little bit more about this adventitia and these peritoneums. As we mentioned, those mesenteries are a double sheet of peritoneal membrane that helps to partially anchor the organs in place. Right? Because while it's important that our small intestine be able to move around to digest our food, right? Tubes like that have a magical property. Back in ancient times, there were things called earphones, right? And earphones or earbuds were these little things that you put in your ear that would allow you to hear. Not that different from the earbuds that we have today, but they had a cord coming off of them, and you would have to connect that cord to your Walkman or whatever else it was that you were listening to. And they had a magical quality. You could take these, you could put them in your pocket and instantly take them out. And what would happen to the cable when you pulled them out?
They would be completely straight all the time.
Yeah, exactly. They'd be completely straight all the time. Absolutely. No, exactly. They'd be tangled almost instantaneously. So while we want these organs to be able to move freely, we don't want them too free where they're going to tangle up. And so these mesenteries, and again, mesentery, it's important to remember, is the generic term for these. They're going to loosely anchor in place and provide that passageway for the blood vessels, the nerves, and the lymphatic vessels. And like I said, they cover most, but not all of the organs in the abdominal pelvic cavity. Now, as I mentioned, mesentery is a generic term. Here, we actually see some specific examples. So again, notice, let's make sure we understand what we're looking at here. Here, for instance, is a piece of the liver. And notice, as you can see here, I'll change to a highlighter so I don't obscure it. Here in green is our parietal peritoneum that is lining the wall. But now we can see where it comes off, and then it becomes the blue. That is small as I can make it. No, it's not going to make it smaller. Let's do this again. So we have the parietal peritoneum here, where it then connects to the blue, which is the visceral peritoneum that wraps around the organ. And notice as it wraps around the organ and gets to the other side, those two visceral layers come off to form a parietal layer that then attaches to the stomach. So notice right here, we have a little bit of mesentery. This mesentery anchors the liver to the stomach, and it gets a special name. It is the lesser omentum. Notice that visceral peritoneum surrounds the stomach, and then it comes off the stomach, forming a double layer that drapes like an apron along the anterior surface of much of the abdominal pelvic organs, and then comes all the way back up where it attaches to the large intestine, specifically a part called the transverse colon. And this big, large draping apron structure we call the greater omentum. Notice the transverse colon of the large intestine is also anchored in place by a mesentery called the transverse mesocolon. The small intestines are anchored in place by a mesentery that, notice they just put mesentery here. We are going to more appropriately refer to it as the mesentery proper. So mesentery is going to be the generic term we will use. When we talk about the mesentery of the small intestine, we will call it the mesentery proper. And notice one more thing. Not all of the organs here are embedded within this serous membrane. Notice the bladder is beneath it. The rectum is behind it. A piece of the small intestine is behind it. The pancreas is behind it. Some organs are not inside of that serous membrane, and those are the ones that would have adventitious. Those are the ones that don't move around. Notice we can change the view a little bit. Here again is that great view of the mesentery proper that is partially anchoring our small intestine in place. Notice here, we can see that transverse mesocolon that is holding the transverse colon to the back wall. And notice they've taken that apron, the greater omentum, and they've raised it up. So they've lifted it up. It's not obviously how it normally is in the body. They've lifted it up so that we could see the transverse colon underneath for that greater omentum. And that allows us to see one other thing too. Notice here, this distal part of the large intestine, what is known as the sigmoid colon, is also held in place by a mesentery. And this mesentery is the sigmoid mesocolon. So if you look on your anatomy list, you'll see all of these specific mesenteries are ones you are responsible for. Oh, I love this picture here. We haven't gotten to the anatomy of the stomach yet, but as you can see, the stomach has one concave side and one convex side. The concave side is known as the lesser curvature, and the lesser curvature is where the lesser omentum is located. The convex side is known as the greater curvature, and that is where that big, long, drape-like greater omentum attaches. So the lesser omentum connects to the lesser curvature, the greater omentum connects to the greater curvature. These pictures from your textbook do a really nice job of showing us all these mesenteries, but the other place you can go to see them very nicely is, of course, that virtual anatomy lab from Cosumnes River College. Notice if we go to the digestive system, there is a digestive system chart here we go. This one's a good one. Notice here, we can see a couple things. We can see a little bit, and again, if you're in the lab, I highly encourage you to take a look at this. a little bit of the greater omentum coming off of the greater curvature of the stomach. Notice here they've cut the transverse colon so we can actually see the cut transverse mesocolon very nicely there. Here we can see some of the mesentery proper holding the small intestine in place. And again, there is that greater omentum draping down. And here's that transverse mesocolon cut again. The other place where we should be able to see this really well, I'm not sure where they would have it. Let's see if they, no, that's the rectum. Ah, this is what I wanted. In the classroom, there is this abdominal, pelvic, and thoracic cavity viscera chart. Notice here, they've done a nice job of showing us where that greater omentum would be, where it is cut there, and a little bit of the mesentery proper. Remember that we would identify there. But this is the one that I like a lot, because this one, notice they do a great job of showing us that lesser omentum that connects the liver to the lesser curvature of the stomach. And again, they have that cut greater omentum. So that's not here. Great view of a cut mesentery proper, a little bit of the transverse mesocolon, and we can also see a little bit of the sigmoid colon that would hold it in place there. So these charts, which are in the classroom and are on this site, are definitely places where you would be responsible for recognizing this, as well as the great pictures in your textbook as well. All right, questions on the mesenteries? All right, as we've seen on a couple of these images, not every organ is inside that abdominal pelvic cavity. So if we cheat and go back to the whiteboard and this picture, we can cheat and take our parietal peritoneum and move it up a little bit. And if we do that, we can see this space where some of the organs might be located behind. So some organs are located behind the parietal peritoneum. And these we refer to as being retroperitoneal. Now, what organs might be retroperitoneal? Does anybody know any of the retroperitoneal organs? Kidneys, excellent. And if the kidneys are, what sits right on top of the kidneys?
The adrenal glands.
All right, so that would be the adrenal gland as well. Or what is also known as the suprarenal gland, because it sits on top of the adrenal gland. I mean, sits on top of the kidney, so it's the suprarenal gland. What else? Well, if you saw the picture, you would see I mentioned the pancreas. The pancreas we haven't talked about yet, but it has a feather shape to it. Its head, and it's got a tail, and it's got a body. The head and the body are actually retroperitoneal, whereas the tail actually sticks out a little bit into the abdominal pelvic cavity. We saw the duodenum or duodenum, either of those are acceptable pronunciations. The beginning part of the small intestine. The ileum and the jejunum are intraperitoneal, but the duodenum is retroperitoneal, which wraps around the head of the pancreas. So those two are related. That makes it easy. What else is sitting back there anchored, do you think, to the back wall? How about blood vessels? Do we want the big major blood vessels of the abdominal pelvic cavity moving all over the place?
No.
No, probably not. So the aorta, abdominal aorta, And the inferior vena cava. Oh, that's not an and sign. Are going to be anchored back there. What else? Notice the transverse colon had a transverse mesocolon. The sigmoid colon had a sigmoid mesocolon, but there were other parts of the large intestine, specifically the colon, that didn't. And those are the ascending and the descending. Oops. So the ascending and descending portions of the colon are not. How does bladder, remember, was below, so it's not considered retroperitoneal, but you have hit on something. We need to get urine from the kidneys to the bladder. So the bladder is not, because it's actually below, so it's not behind. But what are the organs that carry urine from the, there we go, Amy's got it, the ureters. By the same token, our stomach is intraperitoneal, but how do we get food into our stomach? Esophagus, excellent. And remember, we mentioned how the ascending and descending colon are retroperitoneal, the transverse and the sigmoid colon are intraperitoneal, but remember it feeds into that back part, the rectum, which is also retroperitoneal. So these are the organs that are all retroperitoneal. And if you have a little trouble remembering which organs are retroperitoneal, then just remember that the organs that are retroperitoneal aren't happy about it. In fact, they're very sad puckers. So if you're retroperitoneal, you're a sad pucker. PP, pucker. All right. Questions on that? All righty, excellent. So let's go back here. Notice again, we can see how this occurs. We see intraperitoneal organs, and even like the liver and the stomach, they can be in line. But some organs may start intraperitoneal, but they're taken behind the peritoneum, and those are our retroperitoneal organs. And again, our retroperitoneal organs are our sad puckers. All right. There have been many ways that we have identified and described the anatomy of our organs. And a lot of times our organ systems, we do the anatomy first and then we do the physiology. However, the anatomy of the alimentary canal is so diverse. I think the easiest thing to do to discover and discuss the digestive system is to work our way through the alimentary canal the same way that that cheeseburger did that you had for breakfast. So much like that cheeseburger you had for breakfast, we are going to follow the mystical, magical journey through the alimentary canal. And of course, that begins where?
Mouth.
In the oral cavity, exactly. And so that is where we are going to begin our discussion of the anatomy and physiology of the digestive system. We will start in the mouth. But let's do that after the break, because we just did a whole lot of histology and a whole lot of vocabulary, and that's always a little draining. So let's take another 15-minute break. It's 2:17 now, so let's come back at 2:32 and we will restart, and I will start the recording at that time. Any questions before we take our next break?
Actually had a question.
Yes.
You kind of said that some people will have the adventitia and some people have the serosa.
No, what I said is some organs will have adventitia and some organs will have serosa. So for instance, hold on, let's go back here. All of these retroperitoneal organs, all the organs that are back here, they're ones we don't want moving around. And since we don't want them moving around, these are the ones that will have the adventitia around them. That fibrous connective tissue holding them in place. So only the retroperitoneal.
Okay.
Have the adventitia.
Okay.
Whereas any organ that is intraperitoneal, is inside the peritoneal cavity. All of these organs are going to have serosa.
Okay.
Okay.
Thank you.
So yeah, so that was the distinction. We hadn't talked about retroperitoneal organs yet, so it wouldn't have been made sense to mention it at that time. So thank you for bringing that up. I'm glad you reminded me. So yes, any organ that is intraperitoneal is going to have the serosa, that serous membrane, that visceral peritoneum as its outer surface. And any organ that is retroperitoneal, or like the bladder, which is under the peritoneal cavity, would be considered to have an adventitia on its outer surface. But again, that's part of the urinary system, so we won't worry about that yet. So for the digestive organs, so notice the digestive organs that we see here on this list are the esophagus, the rectum, duodenum, pancreas. Those are the ones that are going to, and ascending and descending colon. Those are the ones that are going to have the adventitia on them that we're going to care about in this section. Great question. Awesome. Any others? Again, thank you for that clarification. All righty. Then if there are no other questions, then we will go back to here. And again, we'll take our 15-minute break. And since it's 2:20 now, let us come back at 2:35. I will start the recording. All right, see you guys in 15 minutes. Let's go ahead and get started. As I mentioned, we are going to start our mystical magical journey through the alimentary canal, and that begins in the oral or buccal cavity. What defines the anterior border of the oral cavity? Where does your oral cavity begin?
Your lips.
Yeah, absolutely. Your labia, superior and inferior labia. Absolutely, the lips that form that anterior boundary to it. And posteriorly, what is it continuous with? It continues on into what organ?
Pharynx.
Yeah, the pharynx, the muscular tube that is the pharynx. Excellent. As we talked about, its anatomy, its histology is not the same as the rest of a typical alimentary canal, and in particular, the epithelial tissue that helps to form it. Because as we mentioned, we need to be able to withstand the abrasion from the sharp chip or spicy food or hot temperatures or all the acidic wine, all of those types of things. So what type of epithelial tissue did we say line the oral cavity?
Non-keratinized stratified squamous.
Excellent. A non-keratinized stratified squamous epithelial tissue. If we had been able to take 430 on campus, and you know, some of you may have gotten the opportunity to do a cheek scrape, where you take a little toothpick, scrape gently the inside of your mouth, put it on a slide and look at it under the microscope. And remember, we can see the nuclei in those because they are non-keratinized. Also, if we were in the classroom, I'd make you grab the person next to you and open up their mouth and take a peek inside, because when you take a peek inside of someone's mouth, you may notice a little bit of whiteness, often on the gums or on the roof of the mouth or on the dorsal surface of the tongue, you may see a little bit of whitishness to it. That little bit of whiteness is actually due to a slight keratinization that can occur. So if you drink a lot of hot coffee or spicy foods, or tend to eat crunchy things or things along those lines, you can actually get a minor keratinization of some of the tissues in there, and that's typically seen as a whitish appearance to those structures. So that's not uncommon for that to occur. Here we have the pretty picture of that. And again, let's try to relate some of this information to what we know. As we mentioned, both the anterior and also the lateral boundaries are indeed the labia and the cheeks. And what muscles would help to form those structures? What muscle helps me to control and particularly close and purse my lips?
Skeletal muscle.
It is a skeletal muscle. But what was the name of that circular skeletal muscle that wraps around inside of my superior and inferior labia? Guys, didn't think you'd have to go back to all those muscles that we learned back in 430, did you? There we go, orbicularis oris. Absolutely, that orbicularis oris. What about the cheeks? What muscles form the muscular boundaries of the cheeks? In the buccal region?
Buccinator.
Zygomaticus.
Yeah, buccinator muscle. You are correct. The zygomaticus is going to help us to move the jaw, but really the borders of the walls are formed by those buccinator muscles in the buccal region. Excellent. Notice, as we mentioned, what we call the lips, the labia, the flaps of skin, are the entire anterior and superficial structure that can be pulled out. What common people refer to as the lips is the part where you put the lipstick. And that part where you put the lipstick is actually what is referred to as the red margin. In the red margin, this is an area where it's a transition from the keratinized stratified squamous that we have on the outer surface and the non-keratinized on the inner surface. It is indeed keratinized, but it is a very, very thin layer of keratinized stratified squamous epithelial tissues. Because it's thin, we're able to see the connective tissue and therefore the blood vessels in the connective tissue much more clearly in this region that we call the red margin. This is very convenient from a clinical standpoint, because if someone is very cold, if someone is oxygen deprived, if someone is dealing with carbon monoxide poisoning or even has jaundice, we can often see that change in the color in their red margins of their labia. Not only is it very thin, but notice this area has both no hairs and also no glands. Right? Clearly, you don't have any hairs because nobody shaved their lips this morning before class. But how do you know you didn't have glands there?
Because nothing is secreted.
True. So then how do you maintain the integrity of that so that it doesn't dry out and crack?
You have to put on lip balm.
True. And what did they do before they had lip balm?
Saliva?
Live.
Yeah, there you go. You licked your lips. All right, licking your lips to maintain that moisture. Yes, we all have chapstick now. Chapstick has saved the world. But prior to that, you would lick your lips and help to maintain the integrity and the moisture. Excellent. Notice there is this space between the teeth and the labia. This space is known as the vestibule. And notice indeed we have two vestibules. We have the superior vestibule up here and the inferior vestibule down here. And again, if you think back to middle school, when you first started chewing tobacco, that would be where you would take your big stick of tobacco and stick it into that chaw in there. I'm just kidding. But that would be where you would put that. That space would be that vestibule where you would put that. Notice within these, we have a fold in the mucous membrane that helps to anchor those labia in place. These folds in the mucous membrane that help to stabilize and anchor the labia in place are known as the labial frenulum. And notice again, we have two. We have a superior labial frenulum, which stabilize the superior labia, and we have an inferior labial frenulum that stabilizes the inferior labia. So the lips don't completely flap around in space because they're anchored anteriorly in the vestibule. The behind the teeth is the space that we think of as the oral cavity proper. Obviously, these are bounded by the teeth, but as we also know, there are bones that help to form this. So let's start easy. What are the bones that form the roof of the mouth? Let's start even easier than that. What is the correct anatomical term for the bones that form the roof of your mouth? What's it called? What's the skull feature that we commonly refer to as the roof of the mouth?
The hard palate or
Yeah, it's the
palatine?
hard palate, absolutely. Excellent, it is the hard palate indeed. And how many bones make up the hard palate?
Was it 4?
It is indeed 4. Name them. Oh, someone's already, Amy's got one, palatine already. What else? Again, didn't think you'd have to use all this stuff from 430, but we didn't just randomly pull the crap out of 430 out of a hat. There was actually reasons we learned these things. What other bone, not the ethmoid, what other bone? Right, in fact, if we cheat, we can use this and kind of divide it like that. So they're the palatine bone, and what else? You guys are missing the easy one. Well, mandible, I would say, would help to form the floor. So when we're talking about the floor, it would be the mandible. So then the roof would be made by which bone instead?
The maxilla?
Maxilla. Of course, that's only 2 bones, palatine and maxillary bones. So how does that equal 4?
They're paired left and right.
Yeah, exactly. The left and the right. So the left and the right palatine bones and the left and the right maxillary bones. In fact, I can do this easily because I just finished doing this with my 430 class. There we go. This bringing back fond memories to you. You had to learn all those bones and bone features, and we see how the hard palate is made up of 4 bones. The palatine process of the right maxilla, the palatine process of the left maxilla, the horizontal process of the right palatine bone, and the horizontal process of the left palatine bone form the hard palate, forming that roof of the mouth. Notice if we instead look at, oops, see if I've got a good, there is what I want. Sagittal section, notice here we can again see the maxillary bone and the palatine bone, although I can't quite see where they connect, but those there are forming the hard palate, the bony divider between the nasal cavity and the oral cavity. But notice also there are connective tissues that project posteriorly from the hard palate. And what is that posterior projection called?
The soft palate.
The soft palate. And as we also know, that soft palate terminates at that dangly thing in the back of your throat. And what is that?
Uvula.
There you go. Oops. So excellent. Perfect. The floor, I would say, I would agree, is definitely the mandible from a bony standpoint. And then what else helps to form the floor of the mouth so things don't fall out the bottom?
The tongue?
Yeah, I'd say the tongue is a reasonable structure that forms the floor. Oh, hey, look, and I have the picture here as well. So there's the tongue and the mandible helping to form the floor. We've talked about the roof of the mouth and the lateral components of the mouth, all of that oral cavity. Notice also this illustration does a good job of showing us the vestibules, superior vestibule and inferior vestibule there between the superior labia and the teeth, and the inferior labia and the teeth, and the mandible and the maxilla. And then, like I said, we have the entire labia that, again, is edged with the red margin. So again, these are not new structures. These are all things we learned in 430. We may not fully remember them, but they are definitely things I know you learned in 430, no matter who you had. So now that we've talked about the basic gross anatomy, let's talk about which of those digestive functions that we talked about take place in the oral cavities. So we had 6 basic functions in the oral cavity. Which of those occur in the digestive system? Which of those occur in the oral cavity?
Ingestion.
Yeah, let's start with the most obvious, ingestion. In fact, this is the only place ingestion occurs. I mean, I guess technically you could have a feeding tube where solutions are put directly into your small intestine, but I'm not sure that anybody would call that ingestion in that case. So yeah, this is the only place that occurs. What else do we have?
Mastication.
Excellent. So someone mentioned secretion of saliva. Absolutely. Why is that saliva important? What does that saliva do for us?
Isn't it the first process of digestion?
True, but what exactly does it do to help to facilitate digestion? What is its function?
It has enzymes like salivary amylase.
Excellent.
Break down starches.
Absolutely. Chemical digestion starts here, specifically, as you mentioned, for our carbohydrates. With an enzyme called salivary amylase. For those of you who have kids, this is a fun activity you can do for them. Take a paper towel, have them dry off their tongue, give them a little square of white bread and have them put it in the center of their tongue and just let it sit there. As their tongue produces saliva and that saliva mixes with that bread, the salivary amylase will start to break down the simple carbohydrates in that white bread, and they will start to get a sweet sensation, a sweet taste in their mouth as a result of that. Now, it turns out salivary amylase is not the only enzyme produced in the mouth. There is also a lingual lipase. What do you think a lingual lipase breaks down?
Fats.
Yeah. Does that mean if I take a little cube of butter and I stick it on my tongue, or a cube of lard and stick it on my tongue, then let the saliva mix with it, I'll start to taste the alcohols and the fatty acids as it gets broken down? No, Amy said no, why not? You're 100% correct. Do you know why?
Emulsification.
That's a great guess. Absolutely, yes. Emulsification does help in the digestion of fats. But in this case, not only does it not break it down, but when produced in the mouth, it is actually inactive. It doesn't become active in the stomach. Why? Why would going to the stomach make a difference and allow that lingual lip base to become active?
Absorption.
True, absorption can
pH.
occur in the stomach. There we go. Remember, one of the things about proteins is that they're highly sensitive to pH changes. So when the lingual lip base is produced, it's produced in an inactive form. And when it's released into the mouth, it's in this inactive form. And what's the pH in your mouth?
Around 7.
Yeah, about 7, absolutely. And what's the pH in your stomach?
Two.
Around
2 to 1, yeah, 1 to 2, absolutely. So when, as we know, proteins are sensitive to pH changes and temperature changes. So when we swallow that enzyme down into the stomach with the change of pH, it changes its shape and it suddenly goes into its functional form. So it is going to help us to start the process. You guys were absolutely correct. I didn't catch who wrote it. We're not going to complete the breakdown of lipids in the stomach, but it will start in the stomach thanks to this lingual lip base. So even though it's produced in the mouth, it doesn't become active in the stomach. Excellent. I also heard mastication. What's that a fancy word of saying?
Chewing.
Chewing, absolutely. And which of our 6 functions of the digestive system is mastication?
Mechanical.
Yeah,
Mechanical.
exactly. Oops. As I said, I'm sure everybody in here, when they take a bite of food, chews it the 32 times you're supposed to before you swallow it down, doing that mechanical digestion, breaking it up, making it easier. Does the saliva do anything else for us?
Does it provide lubrication?
Excellent. It is going with the mucus of it. Greases the wheels to help to facilitate propulsion. Excellent. And remember, as we talked about in the last section, it contains lysozymes for breaking down antibodies. It contains, for breaking down pathogens, bacteria, things like that. It contains immunoglobulin A antibodies to stop things from getting in. So there's another important functions that way as well. What else? So chemical digestion, mechanical digestion, ingestion. What are the remaining 3? Well, we kind of have propulsion we just mentioned
Would
with the
throwing up-
mucus, and then also with swallowing or deglutition. Swallowing, moving the food from our oral cavity to the pharynx and beyond. Excellent. That only leaves 2 more. Does defecation take place in the mouth?
Would throwing up count for that?
It wouldn't be considered a defecation. You have the right idea. Vomiting is a way of eliminating materials from the body that could potentially be harmful to the body, but we wouldn't consider that defecation in that case. That's more of a defensive reaction. So that would occur. So you got the right. We can remove materials that way, but that wouldn't fall under defecation. But that does mean absorption, leave absorption. Does absorption occur in the mouth? Well, when grandpa's having one of his episodes and you have to give him his glycerol pill, where do you put it?
Under the tongue?
Under the tongue, absolutely. There is, again, it's not the primary location, but there is going to be some limited absorption that takes place. Certain medications, alcohol, some fatty acids, things like that can indeed be absorbed in the mouth. So if you think of it, 5 of our 6 functions in the digestive system all have the potential of occurring within the oral cavity. Right, so we have that ingestion, the mechanical digestion, our saliva, our enzymes, that absorption, and that doesn't even include our sense of taste. As we mentioned, your sense of taste, in particular, your enjoyment of the taste of the food, can also play a huge role in helping to enhance and stimulate digestive function. Or the opposite is true as well, right? When you see that beautiful Thanksgiving turkey and you bite into it and it tastes like sawdust, right? That actually has the ability to inhibit. So how something tastes or smells or looks, absolutely, those are things that can affect our digestive function as well. Excellent. Speaking of taste, that is one of the major functions of our accessory organ known as the tongue. Notice, and again, we're not going to have the opportunity to go through every single entire anatomical structure on your anatomy list, but hopefully you figured out that by this time. But notice here we can see, and actually I'm going to go back to that other picture we were looking at before. We can see those arches that we were talking about, and then the fosse, the indentation where our palatine tonsil was located. Notice we saw that on the chart before, but if I remember correctly, I think this picture was pretty good. Notice this picture does a really, really great job of showing those two arches in the oral cavity, the palatoglossal, which goes from the hard palate down towards the tongue, and the palatopharyngeal, which goes from the roof of the mouth down towards the pharynx. And then, as we mentioned, we have this fosse, this indentation in between those two. And that indentation is where we find those palatine tonsils. So we talked about that a bit in the last class, but this is a good reminder of that, because when we were looking at that picture before, I saw how pronounced those arches were, and they do a good job of showing that. So we see that there as well, all of which has nothing to do with the tongue, but seeing that reminded me of it. Basically, the tongue has two main functions. Well, two main parts, let's say it that way. As you can see, the anterior 2/3 is dramatically different from the posterior third. The posterior third, we've already talked about. This posterior third is basically the lingual tonsils, covered with those lymphatic nodules, those tonsillar crypts, helping to provide part of our body's defenses for any food and drink that is coming in through our oral cavity. Whereas the anterior part of the tongue, the anterior 2/3, is responsible for moving the food around in our mouth, helping to present the food to our teeth for that mastication, right? It's going to play a role in taste, obviously, as we talked about, can enhance. And of course, we also use it for speech as well. So those are things that it is important. Notice the surface of the anterior 2/3 of the tongue are not smooth. There are basically four main types of projections that stick off of the surface of the tongue. The largest of these are the circumvallate papillae. These circumvallate papillae are these really large papillae that line up in a row, helping to form the boundary between the anterior 2/3 and the posterior third. These are, like I said, are called the circumvallate papillae, although more and more you will see in the literature where they just call them vallate papillae. So you'll see that as well, and I will accept that. Like I said, these are the largest papillae, and they contain taste buds that help us to provide our sense of taste. But they're not the only ones who have taste buds on them as well. Notice over the surface of the tongue, there are these prominent, enlarged, almost mushroom cap looking extensions that project out off of the surface. And all of these, because of their mushroom cap appearance, are what are known as the fungiform papillae. Both the fungiform and the circumvallate papillae contain our taste buds. Now, if you've ever been in an elementary school or a kindergarten class, one of the things that you've seen in there is they have all the pictures of the 5 senses on the wall, right? They have the skin, and they have an ear, and they have an eyeball, and they have a tongue. And on the tongue, you see this great map where you see that you taste sweet here on the top, and salty is over here on the side, and bitter is here in the back. And we have this taste map. And pretty much every kindergarten class I've ever been in has one. But you know where else you see a taste map? Anybody seen a taste map someplace else? I'm asking the question, so obviously it must be somewhere else. Anybody else experienced one of these taste bud maps someplace else?
Wineries.
Yeah, where I saw mine wasn't quite, you have the right idea. It wasn't quite a winery, but it was a Williams-Sonoma. Because at the Williams-Sonoma sells a Riedel wine glass. It is a brand of wine glasses where the average wine glass costs about $30 per glass. Why? Well, 2 main reasons. One is each one is tailor-made for the specific wine in 2 ways. First, they have the right shaped bulb to them so that as you swirl your wine, the right amount of oxygen is mixed with it. But the second reason they're so expensive is the lip of it is shaped properly so that as it pours the wine into your mouth, it hits all of those specific regions of your tongue for those specific taste sensations, which maximizes your enjoyment of the wine. And since it can do that, is it worth absolutely every penny of it?
No.
No, we don't all have Riedel wine glasses at home.
What if you don't drink?
Well, then shame on you. You need to start. No, of course not. It is total crap. Because while we all have taste maps, again, this is one of those fun things you can do with your kids. We all have taste maps. Nobody's taste maps are the same. Again, this weekend while you're studying this material, sit around the table with your family, have a bunch of paper towels, and have a bunch of little cups, right? Wipe off your tongue, and then take a Q-tip and dip it in some lemon juice and rub it over the surface of your tongue. And as you do that, what you'll notice is you don't taste the sour everywhere on your tongue. You'll taste some of it here, and maybe some here, and some here, and some here, and some here. Then you rinse your mouth out, and you do the same thing with sugar water, and you can taste sweet here, and taste sweet here, and taste sweet here, and taste sweet here. And then you can do coffee for the bitter. You can use MSG for the umami. If you're sensitive to MSG, you can use like the juices from red meat or mushrooms, cooked mushrooms if you're a vegetarian. And you can do all these taste maps, and everybody's got a taste map, specific areas of their tongue that are specific to specific taste sensations, but everybody's map is different. So that whole Riedel wine glass thing is total BS. When you saw it at the fancy winery, that was BS as well. There is no standard taste map to the tongue. All right, so drink it because you want to or because you like it. Or better yet, don't drink. Do drugs. All right, excellent. Now,
I have a question kind of
Yes.
about that.
Yes.
How come when you plug your nose, it can impact how you taste? Like when you're a kid and you don't want to eat something and you
It's a
plug
great
your
question.
nose.
Because at our taste sensation and our smell are both chemical perception. We are perceiving chemicals that are dissolved in fluid. For our smell, it's ones that are aerated. And for obviously our tongue, it's the ones bathing the surface. And the two are very intimately related to each other. So much so that while holding your nose while you're eating something that you think perceive as being gross, has an effect, or your sense of smell your sense of taste is dramatically decreased when you have a cold and your nose is congested. But one of the ones that I think is actually the coolest example of this is funky cheese. Is there anybody here in the class who likes eating funky cheese or is at least willing to admit that they like eating funky cheeses?
Yeah, like blue cheese.
No, I'm
Yeah.
talking about the really funky stuff, even beyond blue cheese, you know, like Limburger and those kind of things.
How can you
A
get
couple people, absolutely. One of the things that you'll notice to the people, a lot of people are turned off by it because they smell it. And when they smell it, it smells really, really gross. But when they taste the cheese, it tastes very differently. And one of the reasons for that is so interesting is that, as we talked about, your sense of smell affects and influences and changes your perception of taste. But the smell of the cheese coming from the front of the nasal cavity, whereas when you put it in your mouth and it's coming up from the back of the nasal cavity, we perceive those two smells differently. So how the cheese smells to us from the outside versus the inside of our oral cavity is different. And so you perceive it differently when it's inside your mouth than you're tasting it. The olfaction, the sense of smell, and gustatory, those two tend to go hand in hand in research, is a really, really interesting field of study in neuroscience. It wasn't my area of specialty, but I know a couple people who did that, who does study that. I had some good colleagues and friends that did that, and they're a funky bunch. It is a really, really interesting field of study. Could be typically with the Dijon mustard, it's the vinegar, the acidity that of it that, again, when it's inhaled internally, can cause that burning sensation from that as well. Now, notice as we talked about, those fungiform and circumvallate papillae are the ones that provide us with our sense of taste throughout our entire lives. But there are two other types of projections on the tongue as well. Notice here on the lateral aspect of the tongue are these ridges, kind of make it look like the edge of a leaf, which is indeed where it gets its name as a foliate papillae. What's interesting about these foliate papillae is they tend to, in adolescent, and when we're infants and adolescents, they tend to house taste buds associated with milk fats and milk sugars. Why might it be convenient as an infant and an adolescent to have special receptors that are sensitive to milk fats and milk sugars?
It's useful for breast milk.
Yeah, because that's the primary diet as an infant. Absolutely. Again, as I mentioned, these gustatory researchers, neuroscientists, are an interesting bunch. One of the things they found is that some people during infancy and adolescence have more taste buds on their foliate papillae than others. And so they tend to be more sensitive to tastes. And it is believed that this may be one of the things that leads to certain adolescents being much more picky about their diets. Right? There's some kids that would rather have their left arm ripped off than eat a sprig of broccoli or to eat a mushroom. A mushroom to them is the grossest thing ever. You as an adult, right, that mushroom, yeah, maybe it tastes a little bit earthy, but it doesn't have a strong, powerful taste to you. But because of these foliate papillae and their receptors there, they may be extra sensitive to some of these tastes. So some of these really, really picky, really finicky eaters may be super tasters in their foliate papillae. And again, it's typically something that we grow out of because as we age, those taste buds go away. Now notice that does leave one more type of papillae, the filiform papillae. Notice here our tongue, all the other parts of the tongue look smooth. All of this part in here looks smooth to our appearance. But instead, it's not. This surface is covered with these very fine pointed papillae. And these fine pointed papillae are what are known as filiform papillae. For many of you who have ever been, how many people here have been licked by a cat before? Anyone here been licked by a cat before?
Yes.
Excellent. What does that feel like?
Sandpaper. Kind of.
Yeah, it's very rough because those cats have very pronounced filiform papillae. So based on that, what do you think the function of these filiform papillae are?
Some kind of protection layer.
Some of it is definitely touch, right? If I put a marble in your mouth, assuming it was clean, right, so it didn't have any taste on it that way, would you be aware of the fact that you had a tongue, you had a marble on your tongue?
Yeah, definitely.
Yeah, because of the touch of it. If I had put it in the fridge first, or if I'd put it in the microwave first, would you be able to tell the temperature of it?
Yes.
Yeah, so definitely provides some temperature, some touch sensation, but it's more than that too, right? It was real rough when your cat licked you, but what if instead your cat's drinking water? Does it drink water by putting a straw in the water and just sucking it out?
No, it like picks up the water in
Picks it up
between
with
little
what?
grooves.
Yeah, with its tongue, right? It provides friction. It provides surface area, and that surface area provides friction. For the cat, it helps them to drink. That friction actually, with the surface tension of the water, allows them to pick up water droplets and drink it that way, right? What our cat eats, again, your cat, gets canned food and, lives the plush life, but, cats used to be carnivores, and the things that they would eat didn't want to be eaten. So having a lot of friction on their tongue helped to keep that food in their mouth, right? We don't have those issues. We eat anything you can get through a drive-through. So while many of you have been licked by a cat, how many of you have been licked by your boyfriend before? Does that feel the same? Does their tongue feel the same as your cat's tongue? No one's willing to admit that they've been licked before by a person. Well, I will tell you, no, the tongue is not as rough as, for those of you who haven't experienced it, I strongly encourage it to you. But also that tongue is not nearly as rough as that one as the cat, because while we have these filiform papillae, they're not nearly as pronounced. But they do still give us surface area friction and then tactile sensations, touch and temperature and things like that.
Would the filiform papillae aid in self-grooming as well?
For me?
No, for cats.
Oh, for cats. Yeah, I assume the friction for that helps them to lay the hair down, helps them to move the oils and remove the dander and stuff along those lines. Sure, absolutely.
I mean, I don't know what you do on your weekend, but
I don't groom myself with my tongue. All righty, excellent. All right, spectacular. And I think we've kind of hit on it, but what are the 5 taste sensations? I think we mentioned most of them, but let's make sure we understand them.
Touch, sight, smell.
Well, those are senses. Taste. As I mentioned, there are 5 taste sensations. What are the 5 taste sensations?
Sweet,
Salty.
sour,
Sweet, I heard salty. What else?
Bitter.
Bitter, I saw sour. And what's the 5th? There we go, Maximilian's got it. Umami. Based off of the Japanese word from the Japanese researchers who first identified it. Basically, it means savory. Like I said, that's the sensation you get from MSG, or the sensation you get from the juices from red meat, or the juices from like a portobello mushroom, those types of things, that savory sensation. And again, also remember, as we've talked about, these are chemoreceptors. Oops, that didn't write. Chemoreceptors. Basically, the chemical is a key that fits in the lock, and when it turns the lock, you have that taste sensation. So that glucose gets into the sugar receptor, turns the key, and you perceive sweet. And that's the key. Well, the key is the key, right? Pharmaceutical, not pharmaceutical companies, but food companies, drink companies, Coca-Cola spends billions and billions of dollars each year to find the newest chemical that can get in there and turn the key of that sweet receptor, so we perceive that sweetness without there being sugar, right? That's how we make the ultimate diet drink, right? Make it taste sweet without actually having the sugar in it, right? They just spent billions of dollars. They changed their recipe for their Coke Zero because now it has an improved taste, because they were able to get, a different chemical that's able to get in there and turn the lock. And notice what else is missing from that list. Notice there's no spicy. Why isn't there a spicy taste sensation? Don't we perceive things that are spicy?
Isn't that more to do with irritation?
Partially, absolutely. It's twofold. You've absolutely hit it on the key. First of all, remember the same way that those chemical companies, Coca-Cola is trying to find that chemical that can turn the key. Capsaicin, which is the primary chemical that gives spicy foods their spiciness, their hotness, is like a janitor's key. And what does a janitor's key open?
Everything.
Everything.
Everything. So that capsaicin gets into all of these, turning those keys, causing a huge confusing neural signal that goes to the brain, and that is perceived as pain. But it also stimulates those temperature and tactile receptors that we talked about on the filiform, thinking that it is being damaged. And that's why you get the red coloration of the skin. That's why you get the sweating, is because your taste receptors are being confused into thinking that you've eaten something really, really hot. And so you're being damaged. So that redness comes in, or the dilation of blood vessels to get rid of the heat, the sweating, all of those from those. So it's basically tricking the receptors that are there and giving it that confusing sensation. Awesome. Excellent. Questions on that? True, I agree. It is a little bit weird that it's not classified as a taste per se, but the difference is, like I said, there isn't a single cell that when that cell, binds to the glucose, produces the electrical signal and we say taste. I mean, we taste sweet, right? There isn't a single cell that does that. And that's why it's not considered a taste sensation. It's really a tricking or confusing of the taste sensations that we perceive that way. Lastly, within our tongue, we have a tremendous number of both extrinsic muscles, muscles that start in the tongue and then come out, primarily binding to the hyoid bone. And we also have a large number of intrinsic muscles, muscles that are completely embedded within the tongue. And what is the difference in the functions between an intrinsic muscle and an extrinsic muscle of the tongue? What do extrinsic muscles of the tongue do?
Move it in and
Yeah.
out.
Not just in and out, but up and down and left and right. It moves the tongue through space. Absolutely. So the extrinsic muscles move the tongue through space. So then what do the intrinsic muscles do?
Keep it anchored.
But the intrinsic muscles are completely inside the tongue. So they're not able to move the tongue through space, but what can they do to the tongue? There you go. Noga's showing us. Changing the shape of the tongue, right? Being able to roll your tongue or turn your tongue over or put the ripples on your tongue. They change the shape. Excellent. And of course, how are both controlled? Both the extrinsic and intrinsic muscles of the tongue are controlled by what? A little part of me's dying on the inside.
The nervous system.
Which nerve?
Was it the glossopharyngeal?
Close. Sean's got it. The hypoglossal cranial nerve. Which cranial nerve was that? So cranial nerve 12, the hypoglossal nerve, controls the extrinsic and intrinsic muscles of the tongue. Excellent. Oh, here we see some nice electron microscopy views. Here we see that massive circumvallate papillae. Notice here we see that mushroom cap fungiform papillae. And here we can see all of these more pointy filiform papillae. So notice again, when you look at the surface of the tongue, it looks relatively smooth. But under the microscope, you can see all those fine point-like filiform papillae, giving it that texture, giving it that friction, and again, touch and temperature. Awesome. Here we see the same thing under the microscope. Again, here we see those fine points of the filiform. As I mentioned, in the fungiform and in the circumvallate, and here I'll use black because I think it'll stand out a little bit more. These light colored circles you see on the side. So here we see, there's a big one, there's a big one. These are the openings, these are the taste buds. So here's some more of those fungiform papillae. That chemical dissolved in the saliva comes in, goes into that taste bud, and that is where we perceive that chemical. And notice they're on the lateral aspect of the circumvallate and on the lateral aspects of the fungiform papillae, but no taste buds on the filiform. Oh, here's some better views of the taste buds. There we go. That's nice. Nice big taste buds on the lateral aspect of these fungiform papillae. And here's some really, really good examples of them as well. And these taste buds are where the taste receptors are, and these are nerves. One of the interesting things about these structures, they're connected to nerves, these sensory structures. These sensory structures only last for about 3 days, these taste buds. Because again, if you think about it, you're constantly drinking spicy food or hot food or sharp food. They're constantly being damaged and being replaced. So for instance, if you burn your tongue, you may lose your taste sensation. Or if you eat really spicy foods, or if you smoke, you may lose your sense of taste, or your sense of taste may decrease for a while. But after about a week, when those cells have repaired and replaced themselves, then that sense of taste comes back. All right, so that is the microscopy of it. Let's talk a little bit more about the gross anatomy. As I mentioned, the circumvallate kind of form a boundary between the anterior and posterior 2/3, or the anterior 2/3 and the posterior 1/3 of the tongue. And as you can see also, there's a small indentation between them, between the lingual tonsils and the circumvallate pillae, and that's the sulcus terminalis. Our tongue, like the labia, are anchored in place by a fold in the mucous membrane. And this fold in the mucous membrane is known as the lingual frenulum. So if you notice, we have 3 frenella. We have a superior labial frenulum, inferior labial frenulum, and the lingual frenulum. In some individuals, especially early at birth, this lingual frenulum may actually be too short. If it's too short, that can affect the abilities of the person's ability to move the tongue around in space. That can affect the ability to produce speech. But even worse than that, it can also affect the ability of the baby to latch on and get a good seal to be able to express milk from mom's breast. What do we commonly call that condition where you have too small of a lingual frenulum? Anybody know what the common term for that is?
A tongue tie?
Yeah, it's actually that's what we call it commonly, is being tongue-tied. Now, of course, are we going to get away with a term like tongue-tied in an anatomy and physiology class?
No, because anatomists hate us.
Exactly. So the appropriate anatomical term for having a lingua frenulum that is too short for proper function is called ankyloglossia. And again, you can certainly be certain that that's going to be a term you're going to have to spell on an exam somewhere.
Is there a lip tie also on the superior and inferior? Is that a thing?
You know what? I have not heard of that.
I feel like I heard of that, but I never knew what it was. I just thought of it with the tongue tie, but I'll look it up.
I guess if it was too short, you might not be able to fully close the mouth. And so it could affect the ability to, again, latch on or affect the ability to produce speech, but I have not heard of that one. Of course, if someone has ankyloglossia, what do we do?
They just cut it or snip it. So
Yeah,
it's
exactly. They basically
not as
just cut
tight.
it. They cut it, and when they cut it, then obviously you have more freedom of movement of the tongue. Some cases, maybe a little bit too much free movement of the tongue. There's a lot of speculation that Gene Simmons either had his cut or tore it at some point. And so as a result of that, what is one of his, key characteristics that associated with
He
there,
can
sticks
stick his tongue out really
exactly.
far.
He has that. Gene Simmons has that ability to dramatically extend his tongue, right, to an almost uncomfortable looking appearance for those who've ever seen it. Excellent. All right. Remember also, as we talked about, it's going to produce mucus, and this is where we produce that lingual lip base, which is, as we talked about also, inactive when it is first produced. Does not become active until we reach the stomach. All right. The last major concept I want to talk about for today is to talk about the salivary gland, but this is going to involve both gross and microscopic anatomy. And again, more vocabulary that we're supposed to have remembered from 430. So let's take one more break. So we'll go back to this. I'll ask if there's any questions on this material and on the tongue, and then we'll take our last break and then come back and finish off with the salivary glands. So any questions on any of the material we've covered in this section? All right, then. So let's switch gears. The last major concept we're going to talk about today will be the tongue. We'll take one more break. It's 3:30 now, so we'll return at 3. I think that should give us enough time. Well, let me see what we still have to go over. Yeah, if we don't get to the teeth today, that'll be okay. We'll see how far we get. All right. So yeah, this will be a good stopping point. Let's go ahead and take our last break, come back at 3:45, and at 3:45, we will pick up from there. See you guys in 15 minutes. Let's see if we can scratch some more 430 itches and see if we can remember some of this stuff. Remember back in 430 when we talked about exocrine glands, we basically mentioned that there were 3 main pieces of information we were going to need to know about them. Obviously, the first is the substance they produce. But remember, we also needed to know their structural classification. And what does that mean again? When I say the structural classification of a gland, what am I talking about?
Exocrine or endocrine?
True, that is one example, and you've got the right idea. It is indeed an exocrine gland. Again, so we do have, these are definitely exocrine glands. We do, and endocrine glands function differently. But remember this, the cut structure, you got the right idea. It's basically, it's how it's made, the parts, how it's made, physically what it is made up of. And then, of course, our functional classification is the third thing we need to know. And what do I mean by the functional classification? And you're right, multicellular and unicellular would be one of the ways we would tell things apart structurally. Absolutely. Anyone remember what functional classification means?
Was
Is
it what-
that, I don't remember what it is, but like apocrine and something
Yeah.
like that.
Merocrine, apocrine, and holocrine. Absolutely. Those are the three functional classifications. And they are basically how they release their secretions. All right. So here we are going to talk about our salivary glands. I notice we have 6 total, 3 pairs, so 3 on each side. And that's what this illustration here does a very, very nice job of showing. The largest of these, the most superficial of these, this one right here is the parotid gland. Basically, it is right here just anterior to your ear. So we have that one there, and it's very superficial, that one there. We also have one basically located directly under the tongue. Someone was during the last part, Maximilian was saying about how he had a swollen salivary gland that could have easily been this one right next to the lingual frenulum because it is just deep to the tongue. And the third one, the deepest, is this one down here, and it is the submandibular salivary gland. So we have those 3 glands, 3 on each side. They are going to produce some component of saliva. So collectively, obviously, they are going to produce saliva. But saliva actually has 2 main parts. Saliva is serous fluid. That serous fluid, as we've talked about, is what has the antibodies, ions, wastes, other things like that that are within the serous fluid, and it gives it the watery component to it. But as any good older sibling knows, you are required by law to terrorize your younger siblings. And one of the best ways to do that is to kneel on the chest of your younger sibling and dribble just the tiniest bit of saliva out till it almost touches their nose, and then you suck it back up. Now, obviously, to be able to do that, it can't just be watery fluid. So the other main component of our saliva is mucus. Oops, mucus, like that. And of course, as we know, the glands don't actually produce mucus. They produce a special protein that hydrates and becomes mucus. And what was that protein that when hydrated becomes mucus?
Mucin?
Mucin, excellent. So these 3 glands are going to produce some combination of serous fluid or mucin. All right. Now, that means they're going to have different types of secretory structures. So they're going to have different types of structural classifications. But the good news is that all 3 of them have the exact same mode of secretion. Right, they all have the same functional classification. And that functional classification, as someone mentioned earlier, is eccrine. Pardon me, not eccrine. That's the sweat gland. Merocrine. What does the merocrine mode of secretion mean again? What does it mean to use the merocrine mode of secretion? Is that the one where the cell ruptures and releases all of its stuff? No, that was
No.
holocrine. It's not the one where the front pinches off and releases. That was epicrine. So how does a merocrine mode of secretion work? How does a merocrine gland release its substance?
Was it
Into a duct.
Yeah, into a duct, but how does it get it into the duct?
vesicles?
Yeah,
Oh.
it releases via exocytosis. It takes the secretion, bundles it up into a vesicle, brings it to the plasma membrane, releases it via exocytosis into a duct and out of the body. So the good news is, no matter which of these three I point to, if I ask you for the functional classification, you don't even have to figure out which gland it is. You know it's going to be a merocrine mode of secretion. So let's figure out the other parts. Let's look at the structural classification, and let's talk about the substances they are going to produce. As I mentioned, the parotid salivary gland is indeed the largest, just inferior to the zygomatic arch, just anterior to the ear. It produces primarily serous fluid. Like I said, this contains the salivary amylase. This contains the antibodies. This contains ions and wastes and the lysozymes and all those other components that are in the watery substance. Now, these are all exocrine glands, so they all have ducts. And if you notice, the pathway of this particular one's duct is kind of interesting. It comes out the anterior part of the gland as a single duct. It goes over the masseter. It actually pokes through the buccinator muscle. And then notice right here in the vestibule, right next to the second superior molar, is where it releases its secretion. Sometimes when you eat something really sour, you kind of feel this sucking sensation in your cheeks. That sucking sensation in your cheeks you feel from that sour material is actually that serous fluid being drawn into your oral cavity to neutralize the acidity of that sour, candy or gum or whatever it is that you're eating. So that sensation is actually the movement of the serous fluid through that duct. And notice it's a single, long, thin duct. We have two things we can be making: serous fluid or mucin, mucus. Do you think a thick, viscous mucus is going to be able to travel that long distance in a single duct and successfully get through it?
No.
No. So just looking at the duct should be a dead giveaway that all it produces is serous fluid. The good news about that is we can tell that when we look at this histologically. Here is what the parotid gland looks like histologically. Now I'm going to put a little whiteboard here so I got some place to write. As we know, these are exocrine glands, and exocrine glands are made up of ducts. And what do the ducts do again?
They deliver the secretion out.
Yeah, they're basically a tube, a PVC pipe that doesn't change the secretion or modify or add anything to it. It's just a passageway to get out. So exocrine glands are made up of ducts and of secretory structures. And if you remember correctly, from 430, there are two possible shapes for our secretory structures. Anybody remember what those two shapes are?
Was it round and like linear looking?
Yeah, I like that. round and linear. The round ones, the ball-shaped ones, were called acini. Right? Those are the round ones. And the linear ones were tubules. Now, notice, again, one of the things we often pay attention to or don't pay attention to when we look at things histologically is the color. Because color can be meaningless except when it's not. And this is a perfect example. Notice as we look at this, the first thing that should jump out at you are the ducts. Ducts are typically cuboidal or columnar shaped cells that are rolled up into tubes to carry the material out. So here we can clearly see the ducts. The rest of these things are the secretory structures. Now notice as we look at these secretory structures, they're all pretty much uniform ball-shaped structures. And they're very dark in color. We've seen things like goblet cells that make mucin. And typically the things that make mucin, does stain bind to mucin?
No, it's actually
No.
clear.
Yeah, so typically when we see it under the microscope, it's very, very clear, right? These are definitely not clear. So based on the fact that they are dark stained, do you think they make mucus or do you think they make serous fluid?
Serous fluid.
And since they all tend to be relatively uniform in shape, after all, if you take a ball and no matter how you cut it, it's going to be round. Is the same thing true for a tube? If you take a tube, does it matter how you cut it? Is it always going to look the same? No.
No.
Right? Think about it. If you cut a bowl of spaghetti in half, would you just see a whole bunch of perfect ends, circular ends of the spaghetti with a bowl of spaghetti like that? No, some you're going to cut longitudinal, some are going to be cut oblique. They're going to be at all sorts of different angles and stuff. So the dark stain tells us these make serous fluid. The relatively uniform round shape tells us it's an acini. And then the last thing we need to do is we need to count the number of ducts and the number of secretory structures. And what number do we have to get to?
2.
2,
Yeah.
right? Do I have 2 or more ducts?
Yes.
Do I have 2 or more secretory structures?
Yes.
So based on that information, what is the structural classification of this gland?
Is it a branched acini?
Close. Remember, branch would be 1 duct and 2 or more secretory structures, but this is 2 or more ducts and 2 or more secretory structures. So that makes it a
Compound tubular
Are these tubes?
or alveolar compound.
Yeah, acinar, or you are correct. Alveolar or alveoli is another term that is acceptable for that as well. All right. I think I'm going to lose this if I change the screen, but I want to bring us back to the whiteboard to summarize this. So for our salivary glands, the first one is our parotid. And for that parotid gland, Again, let's think about the things we know about it now. Where is it located?
Inferior to zygomatic arch and anterior to the ear.
I would accept either of those, or we could put both. I like that. Where does its duct release the secretion?
The vestibule at the second molar.
Superior vestibule. Excellent. Let's go easy. What is its functional classification?
Was American.
Yep. And what does that mean again?
It releases its secretion through exocytosis. Exocytosis.
Perfect. What is its structural classification?
Was it compound alveoli?
Excellent. Or again, you can say acini. Both of those are acceptable. And substance it produces.
Serous fluid.
Right. Excellent. That is everything we have not learned about the parotid gland. And so the nice thing about that is not only could I show you a picture of a parotid gland and ask you all of this, but I could also show you the histology slide of a parotid gland and ask you all of these questions, and you'd be able to answer that. So if I showed you this on the exam and asked you to identify the gland, you would say what?
The
Parotid.
parotid gland.
Bingo, absolutely. All right, what would identify the structure?
A duct.
Yeah, identify the structure.
Secretory structure.
Yeah, although we could probably be more specific, right?
Acini.
Yes, and I. Substance it produces.
Serous fluid.
Serous
There
fluid.
you go. Mode of secretion or functional classification.
American.
There you go. See how easy that is? Again, maybe a little challenging because we've only looked at one, but let's look at our second one. Our second gland is the sublingual salivary gland. As its name states, it is under the tongue. Right, floor of the mouth, under the mucous membrane. And notice something different about this one right off the bat. Notice right off the bat, unlike the parotid gland, which had one single duct, long duct coming out of it, notice this sublingual gland has many very short ducts coming out of it. Saliva needs to have two components to it. All right, serous fluid and mucin. Multiple ducts that are very short in length. Is that going to be easier to move mucus, or is that going to be easier to move serous fluid?
Mucus.
Mucin.
Mucus. Yeah, so, well, again, and remember, by the time it's released, it's probably been hydrated. So by the time it's coming through the ducts, it's probably mucus at that point. So you're right, it's mucin when it's released, but by the time it gets into the duct, it's probably hydrating and becoming mucus, which is thick and viscous. So we want to make it as easy as possible to get in there. So not surprisingly, its anatomy tells us everything about it. It's going to primarily produce mucus, helping to neutralize and maintain the acidity of the mouth, helping to lubricate, to form that slippery substance, that bolus that we're going to be able to swallow down. And notice it has numerous ducts not just one, but numerous, all lateral to the lingual frenulum. Notice real quickly, not right next to the lingual frenulum, but to the side. I'm going to cheat and actually go back a couple pictures. Notice here, when we look underneath the tongue, these ducts to the side, these are the sublingual ducts, the openings of the sublingual ducts. This we haven't talked about yet. And remember, our parotid is going to release its secretion right into this space here. So the parotid into the vestibule here, and these lateral to the lingual frenulum, these are the sublingual. Oh, I had that picture here. I didn't have to go back. See, sometimes I'm more clever than I know. Boom. Look how different this looks. Again, right off the bat, the most obvious thing you should be able to see are the ducts. Cuboidal or columnar cells rolled up into tubes. But what do you notice about these secretory structures?
They vary in shape slightly, so they're probably tubular.
Exactly. Notice this looks like a tube that has been cut in cross section, whereas this one looks like a tube that has been cut longitudinally, right? Notice this one here, you can actually see it's been cut on the curve. You can actually see the lumen as this one's been cut on the curve. I'm going to go ahead and erase that so you can look, so you can see that curve to it. This one's more oblique. So notice these are much more irregular in their shape. So that is something that is a dead giveaway. We have that irregular shape. But what's the other dead giveaway on this one?
The color.
Yeah.
Brighter color.
And more specifically, the lack of color. That whiteness to these means that these cells are just chock filled with what?
Mucus.
Or remember, when it's inside the cell, it's still mucin. So these are basically mucin producing tubules. And let's do the math while we're in this part. How many ducts do we see?
Two plus.
Yeah, really, more than one is all we care about. More than one, right? Two or plus. How many secretory structures do we see?
More than two.
More than two.
Yeah, two or more, absolutely. So what do you think the structural classification of this gland is?
Compound tubular.
Excellent. So let's put it together. Let's do the same thing we did before, only this time for the sublingual. How would you describe its location?
Under the tongue.
I like that. Excellent. Where does its duct singular? Did it just have
Plural.
one duct? No, multiple ducts. Multiple ducts release their secretion where?
Under the tongue.
Yeah, under the tongue is good. Let's say under and lateral. All right, so it's under the tongue, but lateral under the tongue. Let's do the easy one. Functional classification.
Miraquin.
What is its structural classification?
compound tubular.
And what is the substance it produces?
Mucus primarily.
Oops. Excellent. Perfect. Questions on that?
The dark colored ones in the histology slide, there were like a few cells. Are those the ones that produce serous fluid?
So you're talking about like here?
Yes.
Yeah, so there will be some cells in here that produce serous fluid, and that could be what those are. But these could also be ones that were just cut at so much of just a thin slice, a thin angle of it, that we might not be seeing the interior of the cell enough. Like notice clearly this one, we can see where we just got the edge of the cell. So only a couple, I mean, the edge of the gland. So only a couple of the cells are really noticeable, whereas the rest we're not seeing inside of. So some of those could just be sections like that. But you are right in that there will be cells in here that do produce some serous fluid, but the vast majority, 95+% are producing mucin.
Okay, thank you.
Yep. And that brings us to our third gland. Our third gland is the submandibular gland. Notice it's the one that is deepest. And again, its name basically tells you where it is. It is deep, deep in the floor of the mandible. So it is inferior, posterior, and lateral of the sublingual. I'll say that again. It is inferior, posterior, and lateral to the sublingual. So it's much deeper in the pocket down there. This picture may be a little tricky to see what's going on, but if you look closely, it has but a single duct that actually comes out, and that is the one that releases its content right next to the lingual frenulum. So here are the two openings to the two sublingual ducts. I mentioned their location, single duct, little bit longer, not quite as long as the parotid, but a single one. So not surprisingly, it is going to contain some serous fluid in it, but it's also going to have a mix of mucin in it as well. This one's going to be a mix of both. I mentioned this location because some individuals are able to contract their mouth and or their jaw and throat muscles in such a way where they are actually able to force some of the secretion of the submandibular gland out those openings and actually project it into space. What do they call that?
Leaking.
Yeah, that's the only term I've ever heard for it. I don't know if there's a true anatomical term for it, but yeah, that ability to be able to express that out, that's the term that I've heard for it as well. I haven't heard any others. So yeah, that ability to be able to express that is this one here. One of the other points I want to emphasize is that while this is not the largest of our glands, remember the parotid is the largest of our glands, this one produces the majority of the saliva. Something like 70% of the saliva is produced from this submandibular gland. Now, I am telling you that this submandibular gland produces both serous fluid and mucin. So what can you tell me about it histologically?
It'll look like a combination between the parotid and then the sublingual.
Yes, exactly. It is going to actually say it that way. It is going to contain both mucin tubules and serous producing acini or alveoli. So yeah, it's going to be a combination of both. And lo and behold, when we look at it histologically, now we're seeing not one thing, not two things, but three things. Again, dead giveaway, obvious, easiest thing to see is again the ducts. In fact, there are two ducts here, one duct there and one duct there. So we see our ducts here. But now notice we have some dark stained ball-shaped structures. But we also have these light stained, more irregular shaped structures. Of course, the light colored irregular shaped structures, what are those?
That's the tubular mucins.
Yeah, the mucus tubules. Right, and then the dark stained ball-shaped structures, what are those?
Acini.
Yeah, serous producing acini or alveoli. Excellent. So notice now we can see it really is a combination of the two. And that's going to allow us to easily fill out the rest of our chart. So again, it makes the majority of our saliva. But once again, let's answer all the questions. Where is it located?
Inferior, posterior, and was it deep?
Yeah, I like that. Inferior, posterior, and lateral to the sublingual. Of course, if you don't know where the sublingual is, that doesn't necessarily help you. So we could also say that is located deep in the mandible. I would accept that as well. Of course, I'd want you to spell it deep and Johnny Depp. Oh, I did it again. There we go. Excellent. Where does its single duct release its content? Where did its single duct express in the oral cavity?
Under and medial the tongue.
Okay, I like that. Or we could also say it is also right next to the lingual frenulum. Excellent. What is its functional classification?
Was American?
Yeah, they're all American. Excellent, that makes it easy. What is its structural classification?
Compound tubule acini.
There we go. Compound tubulo acini or tubulo alveoli. Either of those are acceptable. Put those together. And of course, what is the substance it produces?
Both mucus and serous fluid.
And there you go. Our three paired salivary glands. We have now identified them, know where they're located, know where they release their secretion, and we know the answer to the big three questions. What is their functional classification? What is their structural classification? And what do they produce? Questions on that? All right, excellent.
I actually had a question.
Oh, yes,
Sorry.
please. Please.
Is this the only place where mucus is made?
No, goblet cells inside of our mucous membranes produce mucus. So those are single cells. There are mucus tubules in our tongue, deep in our tongue, that produces mucus for our mouth from our tongue. There are other places where we produce mucus, nasal cavity, airways. And so but I think I know where you're going with this. Most glands that produce mucus are tubular in shape, if that's what you were going to ask. The secretory structure tends to be tubular or single-celled cells, single-celled glands that produce mucin.
Okay, I'm just wondering because I tend to have a lot of mucus, but it might be because I smoke a lot. So I don't know.
It could. That definitely could be something that irritates and dries out your oral and your nasal cavities, and so that could, your mucous membranes could be trying to combat that by overproducing or trying to overproduce to compensate. Yeah.
Okay.
All righty. Of course, the key to that is to switch to edibles. All right. All right. Excellent. Any other questions? All right, we did fall a little behind. I wanted to take our time to go through this clearly. So the one thing that I will warn you about is we are not going to, we'll talk about that later. Wait, I'm in the wrong presentation, hold on. That's the one I want. We're back in the bone features again. That's tomorrow's lecture. Like I said, our submandibular gland produces about 70% of our saliva. This kind of gets along with the point you were making. The average person produces about 1 liter to 1.5 liters of saliva a day, but there are many factors that can influence that. Things like smoking, but also how much you eat, how often you eat, the composition of the food you eat. All of those things can cause changes in the amount of saliva you produce, how spicy the food is, things along those lines. Saliva is mostly water with electrolytes, antibodies, those lysozymes, mucins and buffers. And again, our goal is to mash and bind that food into a smooth, slippery substance that can be easily swallowed and propelled through our alimentary canal. All right, we'll talk about control next time. All right, what I did want to say though, the last thing I want to say is we are running a little bit behind, and rather than rushing this, I wanted to spend time with the glands because I know they tend to be more challenging. I think the anatomy, the organization, the structures of the teeth, I think are much more straightforward. So you are definitely responsible for the anatomy of the teeth. You are definitely responsible for the types of teeth, where they're located, the numbers that we have both in our primary teeth and our permanent teeth. The one thing that is kind of tricky, so I'll remind you of it now, is that while a tooth is not a bone, this relationship between the tooth and the jaw, either the mandible or the maxilla, is considered a joint. And as I'm sure you remember from 430, when we discussed the joints, you are responsible for the functional classifications of the joints, the structural classification of the joint, and the specific type of joint. And since I know all of you remember this from 430, tell me what they are. What is the functional classification of this joint between the tooth and the jaw? Well, let's start easy. What do I mean by the functional classification of a joint? I love all the blank stares I'm imagining from you guys. For the terrified, oh crap, he really does want us to know that stuff from 430. Yeah, that's why you had a cumulative final exam. We needed that stuff. Remember, functional classification means how much the bone moves in the joint. And how much do your teeth wiggle inside of your jaw?
How much?
They don't.
Not a lot.
They don't, none, exactly. They shouldn't move at all. So does anybody remember the functional classification of a joint that does not move at all?
Is it a gomphosis?
That is one of the terms we're going to need. It's not this one. The one that affects whether it moves or not is a synarthrosis. Excellent. Gomphosis is a good pull, definitely one of the correct terms we need to know, but that is the specific type. The specific type of joint is a gomphosis. And then the structural classification has to do with what type of tissue holds it together. What type of tissue holds the two bones together? Notice if we cheat and look at this illustration here, we can see that there is a periodontal ligament that actually holds the tooth in place in the jaw. And so based on the fact that it's a ligament, what would the structural classification be? It's late enough in the day, I'll give it to you. Fibrous. So notice on the exam, I can have a big fat arrow pointing right here. And I can ask you these three questions. What is the functional classification of this joint? What is the structural classification of this joint? And what is the specific type of joint? And since those were things we emphasized in 430, I will remind you of them here. But you're also responsible for the rest of the anatomy as well. So what I encourage you to do is look at that between now and Thursday. If there are any questions, I'll answer questions about it at the beginning of class, but then we're going to jump past the teeth and move on to moving out of the oral cavity and the process of that next. All right. So like I said, definitely look at this over. This is definitely material you're responsible for. And then if you have any questions, I'll answer. But I think this part, other than the joints, which I know people sometimes find tricky because of the terminology, I think it's pretty straightforward. All right. Questions on any of that? All right, excellent. Look at that. We finished 5 minutes early. Yay, us. All right, guys. I know it's a long day, but like I said, we got a lot of material to cram in these last 3 days, in these 3 days of lecture for this section. So expect more of the same on Tuesday and on Thursday. So I will see you guys in 2 days. All right, see you on Tuesday. Why do I keep saying that? See you on Thursday. I will see you in 2 days. Thursday we have class. Be a class on Thursday. All right. Any questions?
Professor, are we going to be able to look at our exams?
Yes, thank you. I will do that right now. So let me go ahead and end the recording.